Oi?
Opa, começou. É, então, nós temos uma audiência ampla aí ao redor do globo. E, bom, então vamos começar, né? Isabela, está pronta?
Então,
Ok.
deixa eu só
É,
compartilhar minha tela. Oi.
tá, pode compartilhar.
É, aí, se eu tossir, se acontecer qualquer coisa, vocês não perdoam.
Desculpa, eu não escutei.
Se eu tossir, se acontecer, assim, qualquer coisa, só
Normal.
em inglês mesmo, né?
Tá, apareceu sua tela aqui.
Deixa eu colocar. Vou pegar meu cabelo. Hum,
Ótimo.
Bom que eu vejo a minha tela. Pode começar?
Só um minuto. Ahn, Bom, vou começar em português porque a coretadora, a Kira, ainda não chegou. Tudo foi preparado para realizar a defesa em inglês, então a Izabela vai agora fazer a apresentação em inglês também. E depois, pelo menos enquanto a Kira não chega, né, ela disse que deve chegar mais para o final da defesa, a gente pode realizar a arguição em português, se a banca preferir. Mas a hora que a Kira entrar, daí eu vou pedir que todo mundo passe a falar em inglês. Então, bom dia a todos. Eu quero agradecer a presença da banca, então deixa eu só apresentar. É, Anderson, começar pelo pessoal de fora, então Anderson de Sá Pinheiro, da Universidade Federal do Rio de Janeiro. Anderson, muito obrigado. A Luciana Márcia de Oliveira, do INRA, na França. Lu, muito obrigado por participar. Os membros internos, a Simone Silva Alexandre, minha colega do Departamento de Física. Simone, obrigado pela participação. E a Mariana Torquato Quezado de Magalhães, do ICB, nossa colega da Bioinformática. Mariana, obrigado também pela participação. A minha co-orientadora, Kira Astakova, como eu já disse, ela vai entrar mais tarde, por causa de um compromisso que foi marcado para ela. Então, eu passo a palavra para Izabela, para ela fazer a apresentação. Você tem de 40 a 50 minutos para apresentar, e depois a gente faz uma breve pausa e inicia as arguições. Obrigado.
Obrigada, gente. Vou fazer a apresentação em inglês, como ele já falou. E é isso. Desculpa pela... E aí, se vocês tiverem qualquer dúvida final, ou se vocês quiserem me interromper, entender alguma coisa, fica à vontade também. E é isso.
Isabela,
Oi.
seu som está um pouco ruim para mim. É só comigo que está ruim ou vocês estão ouvindo bem?
Olha, se melhorou.
Tá ruim, sim. Fala um pouco mais perto do microfone,
Eu preciso falar mais alto, né?
ou mais alto ou mais perto do telefone.
Tá. É porque eu estou usando o áudio do telefone, porque o Zoom não está
Uhum.
disposto no meu computador.
Tá.
Deu
Tá.
para melhorou um pouquinho?
Melhorou.
Melhorou.
É porque minha voz está baixa. Eu tô
Tá.
com alergia de tudo isso. Good morning, everyone. Good evening, Luciana. So today I'm presenting my thesis entitled Mesoscopic Models for RNA Self-Dependence and for Oncogene Probe Design with Blocked Nucleic Acids. This thesis is advised by the Dr. Gerald Faber and the Dr. Kira Shtakova. So I have a little brief overview of the contents that I'm presenting. So this is presenting in the alternative formats of the bioinformatics of the department, of course. So I'm presenting a little bit of an introduction. I'm going to discuss the methodology we use it during the procedure. And I'm presenting 2 papers that we published during the PhD and a little brief conclusion. So since I'm going to talk a lot about DNA, and I know almost everyone here knows a lot about DNA, but I'm just going to give overview. But DNA, it's usually found as a double helix with a sugar phosphate backbone and base pairs, nitrogenous base pairs, formed by thymine and adenine, guanine and cytosine. And according with Watson-Crick base pairing, thymine does 2 hydrogen bonds with adenine, and guanine does 3 hydrogen bonds with cytosine. And besides the hydrogen bonding, there is also an interaction between the rings of the bases. So it's called the stacking interaction, pretty much due to the superposition of the electrons and everything. And since I'm also going to talk about RNA, I'm just going to discuss the difference between the 2. So RNA is very similar to DNA, but it has a different sugar phosphate backbone. And it also has, in the place of thymine, we have uracil doing 2 hydrogen bonds with adenine. Although RNA is usually found as a single strand, it also can be found as a double strand. So it can also fold upon itself and as a double strand, same way as DNA. So to describe DNA, RNA, and nucleic acids in general, we have several theoretical descriptions. Here I did a little grouping. So I can speak about microscopic descriptions of the molecule. I can speak about microscopic, so I'm going to describe the atom itself. So this is exemplified by DFT, molecular dynamics, and quantum calculations. And from this, I can get very detailed information such as electronic states and molecular structure and molecular motion. But this is really costly in terms of computational complexity. So I cannot do more than a few base pairs at once. And when I say microscopic, I'm going to describe things like microscopic properties, such as the persistence length of an helix. So this is usually done by the worm light chain model or lattice Boltzmann model. All is not described here, but the newest neighborhood model. I'm going to say a little bit more, a little bit ahead. And I can also talk about mesoscopic models. So this is going to be halfway of these 2 models. So it's going to give me a little bit more detail than a microscopic model, but it still is not going to have such detailed description as microscopic. But in this cost, I have median computational complexity cost, so I can do more base pairs than microscopic probes. And from this, I can exemplify Monte Carlo models and Bishop models. And from this, I can extract dynamic homing fields and statistical and thermodynamics information. So besides the whole DNA structure and everything, DNA can undergo a process that we call denaturation. So denaturation is described as the breaking of the hydrogen bonds and the separation of the 2 strands. So when I say that, DNA can be denaturated by, by a force or by a changing pH, by a changing solution. And it can also be denaturated by what we call thermal denaturation. So if there is an increase in temperature, we are going to have separation of these strands, and what we call the melting temperature is the temperature which half of the strands they form double strands, and half are at single strand in solution. And this thermal denaturation is very important for several processes, and I have just a few examples here. So this is used as I forgot the word, but as a reference and polymerase chain reaction and sequencing techniques and genetic therapy and diagnostic tools, DNA networks, and digital biological circuits. So it's very important that we can describe this thermal denaturation of DNA. But we also have few models that can do that, of course. And the model I said before, the nearest input thermodynamic model. So most of you can be probably familiar with the model, but this model is going to model every single base pair in the double strand as different sums of enthalpy and entropy. And you can sum over any sequence, and then you can predict a melting temperature. So, but this is really empiric, and you cannot have a really physics aspect of the molecule. There is also the Poland-Schrager models. This is also based in a statistical model, but it's going to model the strand as an open and closed state. And there's also some models that include entropy penalty for the formation of bubbles, but it's not very used, especially because it has few problems with the prediction of temperature. And there is also the model that we use here that is called the Ferreira-Bishop model. And I'm going to talk a little bit more in the next slides. So I said about the Ferreira-Bishop model a lot, but the Ferreira-Bishop model is going to describe the double helix of DNA through two very simple potentials. So I'm going to describe the hydrogen bonds, so the thing that makes DNA coalesce, as a potential, as a Morse potential, that I'm going to show the expression a little bit ahead. And I'm going to describe the stacking interaction between adjacent base pairs as this W here, but as a stacking potential that I'm also going to show. But here I'm showing the Hamiltonian of the system. So I have the kinetic port for each direction. And I also have the Morse potential and the stacking potential for each direction. Although the only direction that actually stretches the hydrogen bonds is the y direction. So that's the reason for this only be in terms of y. So the potentials that I'm talking about. So the Morse potential is going to have this format here as the red curve. So it's describing an open and a closed, no, actually an open, bounded and unbounded state. So if I'm too distant from the other base pair, I cannot bound, I cannot make a hydrogen bond. And if I'm too close, I also cannot do it. So I have optimum distance. And the second potential is just basically a trapped potential of a particle. So the expressions here, I have V in terms of the Y and the stretching of the last base pair. And I have the second potential that is an harmonic potential, common harmonic potential. And I'm also showing a Morse comparison of an AT base pair, so adenine and thymine, doing two hydrogen bonds. So I have smaller potential, a lower potential than for a CG base pair, for example, here in red. So I have deeper well in this case. And we are going to solve, as you remember, don't remember, I can show again, but this Hamiltonian is going to fully describe the system. So we can solve this Hamiltonian through a partition function. And the X part, it's a trivial solution. And the part that actually extracts the hydrogen bonds is the Y part, and this is the part we have some things to address. So if we're going to solve the Y part, so the ZY part, so this is the Hamiltonian, so I'm solving for stacking and for the Morse potential. And just to remind where it comes the temperature is directly related with this beta here. So is related with the Boltzmann constant and the melting temperature of the two flats. But to solve the problem, to solve this partition function, we have to undergo several calculations and several algebraic processes. But to describe the helix as a heterogenic helix is introduced. Okay, it's introduced several matrices. And this matrix can be described in terms of an omega, that is basically the amount of CG in its sequence. And this omega, when you look at this expression, this Z, the partition function in terms of omega, you can see that is really related with the content of the sequence. As I said before, omega is related with the CG content of the sequence. So is, of course, you can compare different sequences and say something about different sequences. But of course, the partition function is strictly correlated with the temperature, and we cannot use as a comparative parameter. But when I look for this omega maximum, that is going to describe the maximum content of a sequence, so the sequence that only contains CG base pairs, I have a correlation with temperature, and this is not biased anymore by the partition function. So I can compare different sequences and their contents, and of course predict melting temperature. So going back to my equation, so from that omega maximum that I said before, we call this as a thermal index, and this thermal index, actually, okay, I'm going to show a little bit, but this thermal index is omega maximum. When you look to the regressions, to the interpolate, when you plot the omega maximum against the temperature, you see there is a linear correlation between the length of the sequence and the experimental TM, so the measured TM. So from this relation, you can extract, so you can predict the temperature using your model, and from this you can compare with the experimental temperature. So we have a way to correlate the temperatures that we predict with the temperatures that experimentalists measure at the lab. So this thermal index is directly correlated with the length of sequence, and from this we call two coefficients, so two regression coefficients, so A0 and A1, and this is directly correlated with T prime, so I'm going to call my predicted temperature. And of course, this temperature is going to depend on the thermal index, and this one also depends on the parameters that you have extracted from the parameter plane. And I'm going to discuss this a little bit ahead, but I just want to show this. We also can extract from the partition function what we call average opening. So it's going to be the opening of each base pair at the sequence. So we can, I'm going to show more figures with this description, so it's better to understand, but we can see how the sequence is going to behave with the temperature. So which base pair is more prone to open and is going to have a wider opening than the others. But I said we can extract the temperature, we can compare with the experimental temperatures, but how we do that? We do that by a parameter optimization. So for example, here I have canonical DNA sequence. So it's composed only of CG and AT base pairs. And from this, I can extract a set of parameters that I'm calling here PK. So I should have that senses here, but it's just going to say, okay, how many parameters I have to fully describe any type of DNA canonical sequence. And from this, I have two Morse potentials. So I have a CG and AT. So GC is symmetric to CG and TA is symmetric to AT. So I have only two Morse potentials. And for stacking potentials, doing the same thing as the symmetric, simplification. I'm going to describe each neighbor in the sequence. So for example, the first neighbor here I have is CGTA, and doing the symmetric equivalence, I have ATGC on the other side. So I have two Morse potentials and 10 stacking potentials to fully describe any sequence of DNA. So this is the parameters that I have to use to correlate with experimental temperatures. And I do this correlation. So from the set of parameters, I can predict this T prime, so the temperature that I predict from the Bishop model. And from this, to do this correlation, we do this by, I have the GIFs I can show at the end, but this is not a really important GIF. So this red polygon here is just going to try to find the minimum in the function. So in this function, I show the Rosenbrock function. So it has clear minimums and 4 points of the surface. So the method is going to try to find the minimum values that minimize the function. So it's going to try and find the minimum of the function. So we are going to do the same for the Bishop model, but we are going to correlate the melting temperature, the experimental melting temperature, so this Ti, with a T prime, so the predicted melting temperature, using the parameters. So the minimization is going to search and space described by the parameters. So in the case of canonical DNA 12, so space 12 dimensions, is going to describe, it's going to try to find the minimum parameters, the minimum set of parameters that minimize this difference. So I'm trying to get the best set of parameters that actually gives me the best predictions for a set of sequences, if it does make sense. So, and also as a comparative parameter, we use this median, the average delta difference between the temperatures, between the predicted temperatures and the experimental temperatures. So for this, I have the best chi-square possible, and I also want to have the best delta Tm possible for the set of sequences. So I have a way to correlate the experimental temperatures with the temperatures that are predicted from my model. But I have a few problems, as I said before. We have to correlate. We need experimental measurements. So, as I said, the model is relying on experimental data. So we need experimental data. So if I have different sets from canonical DNA, I will require, the method is going to require a new parameter extraction. So I have different parameters. So for example, for RNA mismatches, for DNA mismatches, I'm having different parameters. So I have to do all this optimization of parameters again. And I also have to do for different experimental conditions. So for example, for different saline solutions, and for different, for different contests. And of course, for different modifications. And, this is costly. No, it's not costly, but it's like a little bit of annoying to do every time, parameter extraction. But after you extract the parameters, you can predict several quantities. So for example, we can predict the melting temperature. You can predict this average opening. You can predict the entropy if you want, just using the parameters that you extracted before. And in a few seconds, so I did a few calculations. I'm going to show a little bit ahead. So until now, our group have several models of several parameterizations of different nucleic acids using the Bishop model. So we have canonical DNA under different salt concentrations. So we have different Morse base pairs. I'm just going to show the Morse potentials here. So the Morse potential is going to change over different salt concentrations. I also have DNA considering terminal effects. So if I consider a different potential for the base pair that I expose it to the solution, I'm going to have different Morse potentials. So different hydrogen bonding. I have this for DNA mismatches, for DNA RNA hybrids, and I also have this for RNA at high salt concentration. We do not have until now parameterization of the Bishop model for RNA under different salt concentrations in the same way we have for DNA. So one of the projects that I was involved into was this silent and terminal effects in dsRNA, double-stranded RNA. As I said, we do not have this for RNA. We only have a high salt concentration. So for DNA, we have way more information. So our purpose was to model this in the same way we did for DNA. And I'm going just to give a little bit of overview why double-strand RNA is important and why we need to model. So double-strand RNAs, they are present in cells and have a variety of biological functions. I'm just showing here double helix of RNA, so a little bit different conformation than in DNA. Small non-coding double-stranded RNA can mediate neuronal differentiation. They can inhibit the translation of RNA messenger molecules, and they also can be key activators of innate immune response against viral infections. And as I said, double-strand RNA is important, but it's also important to describe how RNA is going to behave under different salt concentrations and different salt conditions. So the helix is sensitive to environmental conditions, such as temperature and salt concentration. So here I show different conformations, slightly different conformations for different salt concentrations. So you see that the helix, it has a different conformation, and therefore this is going to affect the melting temperature. And as everyone is familiar or is not, due to the difference of the sugar, double-strand DNA conforms in a different way than RNA. So from this, they have different higher dependent condensation behaviors and also different flexibilities. So we cannot assume that DNA is going to behave exactly as DNA. And this is important, really important. So RNA tertiary structure is also sensitive to the size of the monovalent cations in the solution. So the way that RNA is going to fold is going to depend also on the concentration of the solution. And the terminal effects, why we wanted to study terminal effects. Why? Why can I not assume that internal base pair, so here, for example, is going to behave exactly as this at the end? So why not? This end here is exposed to the solution. So this can interact in a different way with the solution than this internal base pairs. So in the same way as you did for DNA, this is probably going to have or not a different Morse potential. So we wanted to study this in more deep aspects. So these intermediaries, actually, the terminal effects, they are intermediaries in zyping and non-zyping processes. They're important for the interactions of RNA with proteins, and they're also important for secondary structure rearrangements for the switch function. So, as I said before, we did the same for DNA. So DNA is also going to have a different interaction for terminal base pair. And as I showed in the previous figure, we have different potentials for a CG, for example, here, terminal. Both terminal are in red. So I have a slightly lower potential for exposed base pair. So less stronger hydrogen bond. So, and it's also for AT. We have a comportment is slightly different. So for low salt concentrations, this 80 at the terminal is going to have considerably low Morse potential. So this is probably going to open before, of course, than the internal base pairs. So we wanted to study this in the same way as we did for RNA, as for DNA. So, but we had a problem. We had a different collaboration in 2017, I think it started, and we had some struggles with the data, but we published a paper over RNA at different salt concentrations, but using the nearest neighbor model, because we had problems to deal with the set of sequences using the Ferreira model. Why? because although the nearest neighborhood model do not have a problem with the concentration of the strand, so the concentration of the strand at the temperature was measured, the Parebishop model is going to have a problem. Why? Because the Parebishop model is a single molecule calculation. So I need to have all my melting temperatures at the same strand concentration. Otherwise, I cannot assume that they're going to behave as the same. But we thought actually there is not a reason to actually behave as different because it's a single molecule calculation. So I'm not actually treating strand concentration, but we need to assure this. But I'm showing the graph and going to explain, but we have this problematic data in a way that every single sequence that we have here, so I'm showing each color here is depicting a different sequence. So for example, this blue here, this dark blue here is the same as this dark blue here. This is the same sequence, but measured at different salt concentrations. But this is measured over a range of strand concentrations, and we do not have the same strand concentration for each. I'm showing a graph a little bit ahead, but we needed to have a way to use the Parebishop model comprising this strand concentration. We need to adapt the model to handle different strand concentrations. So I'm showing here. When I group the sequences that I have here for different species concentrations, so here I have, see, you can see here, this is the total strand concentration. So I have going from 1 micromolar to almost 7, 8 micromolar. So if I group this, actually, if I do this, the logarithm, so if I take the logarithm of the strand concentration and round this, I can actually make groups. So I can separate and put different sequences in different boxes. So I can do different groups and say, okay, you're probably going to behave similar as this one. So if I measure the sequence at 1 micromolar strand concentration, I'm assuming, depending on the F here that I use in this equation, I'm assuming that it's going to behave, for example, as the same way as a 1.5 micromolar. So we are going to have different groupings. So this F here, the expression, so let me go again. We extracted the logarithm of the CT. The C0 is just to assume that this is a dimensional. And we round this. So we are rounding the number for the most closest number. So I'm grouping in a way. So, but to make this general, so we introduce this F factor here. So for F equal 1, it's going to be the same, of course. But for different Fs here, I show we went from 0.4 until F equal 5. So the most grouped sequences, so I have the lower number of groups. And for F equal 5, I have the highest amount of groups formed, depending on the F. So if I show here, I don't know if you can also see in the same way as I did. I didn't do an histogram because it's going to be a mess. But you can see here, for example, for this red one, 1.3. So you have almost a histogram. So we are grouping a different number of sequences for different species concentrations. So for example, here, for a species concentration of almost 2.5, I'm going to have a group containing 40 sequences. But if I go from 1.5, I'm going to have lower than 10 sequences per group. So this grouping is going to change as we change the F. So we try a different Fs just to assume, actually just to assure that, to be certain that this F is not influencing anything. Or if F have any influence in the results, which is the best F that I need to use. So actually I'm going to show the regressions a little bit ahead. But so this was the problem, and this was the grouping that we did. I don't know if it was clear. But as I said, we need to parameterize the model for each different condition. So we did a parameterization in the model for each different f. So this means I'm doing an optimization of parameters for every single f. So in this case, 16 different f's and 16 different minimizations. So in the same way as I did for the notation for DNA, I'm doing the notation for RNA. So the different, as I said before, I'm having for DNA is an uracil in the place of a timing. So for this, if I want to treat terminal, I'm going to depict this AU terminal as having a different potential than an internal AU. In the same way, I'm going to do for CG, and then it goes. So I'm calling everyone different, not everyone, but terminals parameters, they are different. So I'm increasing the amount of parameters that I need to minimize. And the question I raised before, which K-square is the best? So which F is going to give me the best K-square? Which one is going to give me, as I said before, the interesting is I have the lowest K-square. So which F is going to minimize better this this K-square? So I'm going here from F 0.4 here until F equal 5. And the red one is considering terminals doing this minimization. And the blue one is considering everyone as the same. Of course, if I consider more parameters, I have a better description of the system. And in this way, I have lowest K-square. But it seems for F equal 5, I have a better description. I have the lowest K-square. But in the same way, if I have F equal 1, is it still good enough? Is it still way too close to F equal 5? So we need to assure which F is the best one, or every F behaves as the same, or anyways. So what we did was to I have to just get, just going to show this. So here, I don't know if everyone can see, but here I'm showing every. I'm going to optimize here a little bit. But for F equal 1 here, I'm showing the A0 and A1. If you remember from the correlation that we do with the experimental temperatures, F equal 1 gives A0 and A1 behaving almost as a linear behavior. And this is the same as we see before for DNA regression parameters. But for F equal 5, although I have a very good K-square, I have this dispersive coefficients. So, for example, for 7.1 salt concentration, I have some regression parameters that are really going beyond the line. So this is not good. If you want to correlate this, if you want to use these parameters for different minimizations, to predict different parameters for different sequences without having to parameterize everything, it's good if you have a well-behaved regression. So I did, I guess, just to show, actually, we are going to have a regression for every F. So we're going to have 16 regressions. So if I show here, it stops again. But if I show here, you can see that the regression parameters, as I increase the number of the groups, they're going to be more dispersed and more dispersed until I get the worst scenario possible, that is F equal 5. So I have very dispersed parameters. So we can have linear parameters for almost every F. But if I increase too much F, so if I separate too much the group, so if I have too many boxes to classify the sequences, this is not going to give me a good classification. So I'm missing out something. So let me go again. I just did this because this presentation is not showing the gifts. So we have this question, which F is the best one? So it seems that F equal 1 still gives me good results and still has well-behaved regressions. But if I show, as I showed in the GIF, different Fs also have good regressions. So Fs also have good well-behaved regressions. So what we did was to do was average. So how much this is changing, the changes, minimizations. So here in black, I have the sum. Actually, I have the median of the parameters over all range of salt concentrations. So, and just compare, I have the lowest F possible in green. So you can see they are really close to my black line. And F equal 1 is also really, really close to my black line. And I have a little bit, although F equal 5 do not give me good regression parameters, we still have good results. So it's still really, I don't know, acceptably close to my median. So showing here for F equal 1. Actually, this is F equal 0.4. I did the same with the GIF, so I want to show how this changed over, but I'm just going to discuss a little bit ahead this whole graph, but I want to change. I want to show how this behaves. So you see that although I have a little bit of dispersion of the parameters and a little change over the Fs that I use, they're still too close, as you can see from the media. So we still have the problem, which F is the best. And the question, actually, the answer is any F is good enough. Any F is going to achieve good parameters. And as F equal 1 gives good regression parameters, well-behaved regression parameters, this is the parameters that we actually use. And I'm going to discuss this, but just going to go over stacking potentials. So we also did this average for the stacking potentials. So I cannot show every single F, three comparative Fs here. So this is going to, otherwise this would going to be a lot. But here in the error bars, I have the changing in the parameters over the Fs. So they're still well behaved, but they're still too close. So I don't have too much, I don't know, highest error bars. But in this case, just for this parameter here, weight BAU. And if I also see the stacking potentials as they behave over the Fs, I have a little bit different, but they're still close enough. They still, they change, but they still keep almost the same. So this is not going to make too much difference when I predict the temperature. So, fast. So I said F equal 1 gives the best regression parameters. So that's the reason we are discussing the most potentials for F equal 1. But here in black, I have the parameters for the terminal base pairs of DNA. Compare. And in gray, I have the internal. So for an internal RNA base pair, I'm going to have a slightly higher Morse potential. But for the terminal, I'm not having this this reduction in the at low salt concentration. So this is more stable than a terminal base pair at AU terminal base pair at the RNA sequence is more stable than the AT base pair in the DNA. So we could not consider as the same as we thought. And also the stacking potentials is the same way as we see for DNA in our other words I can show at the end, but they also behave very similarly over the salt concentration, so they do not have very abrupt changes. Especially only this one has this crazy behavior here, but this is not really a deal breaker. But we also need to compare this with parameters. We need to compare with say with experimental data or otherwise with other calculations. So this Jim et al. Here, 2008 from 2018, it studied the opening that is very similar to the average opening for a sequence containing 40 base pairs for different salt concentrations. So you can see for lowest salt concentrations, it shows more open, so less open states, and for increasing salt concentration and have more closing. Actually, I think it's the opposite. So this is contrary from the average opening as we do. So this area MSF, anyone corrects me if I'm wrong. I'm just doing a little bit of mess in my head. But I think this for high salt concentrations, this is more closed, and for low salt concentrations. And we did the same plot using our data, using our parameters for the whole, every single salt concentration that we had under set. So similarly, as I said, is in the opposite direction. So if I have low opening, so if I have a more closed opening or average opening, this is going to be more open as in measuring in a different way. And we have similar openings, especially with this curve here in the middle. And we also have a similar openings for the salt concentration. So our result is still good enough for F equal 1. So a little bit of an outline. We have a new technique to parameterize the Berdyshev model using multiple strand concentrations, using logarithmic grouping. So now the Berdyshev model can treat different sets of data, having different melt strand concentrations. So we can do measurements that we are not able to do before with this limitation. We still, as we suppose it, there is a little dependence in the strand concentration. So there is a little dependence in the same way. There is a little dependence on the group coarseness, due especially to this single molecule calculation. We have new salt-dependent PB parameters, including the specific parameters for terminals in the same way as we had for DNA. And differently for RNA base pairs, you do not have this important reduction that we see for DNA at low salt concentrations. And as I said, this technical directions show little change with salt concentration. So the other work we did was a parameterization of single LNA modifications in DNA. But what is LNA? I'm going to show a little bit in a little bit, but modified nucleic acids had been very important in the last few years, especially due to their way to improve the stability of nucleases and cellular uptake. So you can actually do oligonucleotide treatments and more stability. In this case, we have an improvement of affinity and kinetics when using modified nucleic acids. I'm showing here all these modifications. They are done in the sugar and the locked nucleic acid. This one here has a sugar modified nucleic acid. I'm going to show a better picture. They also have an improvement in the binding specificity of the base pair. So this is really interesting when you're doing treatment using oligonucleotides. And the locked nucleic acid. Why is called locked? Because I have this Magellan bridge actually locking the base pair in a different conformation. So if I only have DNA, for example, I'm going to have an S-type conformation. So this is the reason why DNA and RNA have different conformations. And RNA, due to this additional OH here and the ring, is going to conformate in a different way than DNA. And RNA does the same. RNA is locking this DNA base pair and same conformation as RNA. And because of this, as I said, we are going to have a different conformation. So for RNA, I'm going to have a more closed state. So I have more narrow major groups and minor groups. And for DNA, I have this more open, there's more wider major groups. But actually for DNA here, I have more wider major groups. And for the RNA, although I have the same conformation from the RNA, I have a different conformation, slightly different from RNA, when I have helix only formed by RNA. But why RNA is being so important? RNA has been studied over the years, and there's a lot of words describing RNA. But why? So RNA shows a greater stability and included in the rest of DNA or RNA. So I have a more higher melting temperatures or more stable duplex. And some studies even have an increase in the melting temperature of 10 degrees per modification. And due to this, I have an improvement in compatibility and specificity towards DNA or RNA. And because of this, I have a better mismatch discrimination, especially when I'm trying to find different base pairs in the sequence or single nucleotide modifications then it goes. And it's also resistant to nucleases and advantageously is compatible with automated synthesis, so we do not need a whole new synthesis processes. And due to these amazing features, RNA has been applied in a wide amount of applications, such as PCR detection and diagnostic tools, antisense oligonucleotides, and microRNA silencing, RNA interferencing, aptamers, DNA enzymes, and LNAs. But why? Why LNA shows this better stability, this increased stability? The first thought when the studies they did was an improvement in stacking interactions. So this was a structure that one of the first studies predicted. So they assumed that there is an improvement in the stacking of adjacent base pairs and therefore increasing the melting temperature of these duplex. And other studies, they said, okay, it's not stacking, it's not anything, but you need to include the hybridized water in the studies of NR. So you need to actually include the water interaction with the duplex. So perhaps it's doing different interactions. And this is one of the figures of this study. So they show here in black, I have the RNA modified sequence. And in gray, I have the canonical DNA sequence. So they say there is not this difference. So I can assume that is a hydrogen bonds or the stacking that is included. And another study, so this from molecular dynamics study. So they included this water. And when they include this water, they say, okay, there is a water-rigid hydrogen bonds due to this has the active oxygen in RNA. If you didn't see, RNA has this extra oxygen as DNA doesn't have. So perhaps this is giving more hydrogen bond potential and is strengthening the hydrogen bonding. So they show four interactions with waters for water-rigid hydrogen bonds. But this is when Kira starts. So we have our group collaborated with Kira a few years ago for a study of predicting oncogenic probes using locked nucleic acids, and they wanted to better predict this melting temperature. And what our group did using the small set, was way smaller set of the modified sequences, was to extract these parameters and compare these average openings. And when they see for a modified guanine, so LNA guanine here in red, and this opening showing red. I have a very different opening, so it's way more open than canonical DC base pair. And I thought this is going to be more stable. And this were the openings, especially due to the stack the result. But they only had modified guanines and modified cytosines. So they have a very strong hydrogen bonds for a G plus C, but a very low, actually for DNA, this is in the average 78.1. And they have a very slow, so 78.5 electrovolts. So this is like very different from the results. It was contradicting too much the results that we had that were already published. And in the same way, they show smaller stacking interactions for modified base pairs, and it completely contradicts the assumption that there is an improvement in the stacking interactions. So of course they need some confirmation or to refute these findings and say, okay, we did it wrong, okay, we are right, and that goes. So what I did was, so this is when I start. So I collected, due to the, I don't know, I think it's easier to do this measurements in the high salt concentration. So the literature had a lot of measurements for rocket nucleic acids, especially due to the popularity. So we collected a very good set of parameters of 306 parameters. So I have 3 papers, and these papers are studying different aspects of rocket nucleic acids. But for the model, I wanted to have the modification in the same context more than once. So some sequence had to be extracted. And I also want to only consider single rocket nucleic modifications. So I do not want to consider, for example, if I had unmodified thymine or adenine here. We did not want to do this minimization. So this set is going to only comprising single LNA modifications and at a high salt concentration. So to do an example of the notation the same way as I did for RNA. So if I have a modification here in the guanine, of course, this guanine is different from a canonical guanine. So I'm going to have plus GC. And for example, here it's a little bit shifted, but I'm going to have A plus T is also a different Morse potential, and of course, different potentials. So for the sequence, I'm going to have 2 more Morse potentials and 4 more stacking potentials. In the totality, for the whole set of 306 sequences, we had 4 more Morse potentials. So besides the 2 that we have for canonical DNA, and 32 more stacking potentials for modified base pairs. And this is increased way too much, or an amount of parameters to minimize. So, but I was successful under minimization. So before our minimization, as we said, we calculated the p-square, and it was almost 5,000 p-square. And actually, Celsius is clear. And for the average melting temperature difference, we had a 2.9. So for some applications, this is still not good enough. So we minimize it, this set. So I considered 3 minimization. So an initial minimization, a minimization considering DNA in the minimization. So I also minimize the chemical DNA, and a minimization to have to generate error bars. So my best results is 1.47. So it's actually almost half of my initial, and my p-square almost 5 times lower than my initial. And the results I have, they are here. So for AT and CG base pair, when I minimize them, they keep almost the same initial for AT and CG. I have a very different here. So This means a locked nucleic acid when put on in the sequence is not affecting geometric neighborhood. So it's not affecting it's not affecting the hydrogen bonds of an AT or CG canonical space. But if I see a modification, so for example, here is a base pair containing a thymine and an alanine adenine, and here adenine and alanine thymine, they almost have the same potential as a CG. So they increase almost from 33 for almost 70, more than 70, almost more than 70. And for a modified hydrogen, I also have a very, very increased hydrogen bonds. So this was confirming our primary assumptions that the stabilization of alanine is due to hydrogen bonding, as we say here. So we have this crazy hydrogen bonding here. So it has almost the same value here, 9. And otherwise, when I look to the stacking potential, so this shaded area is showing the area that almost actually all the stacking potentials should lie about. So let me correct. All the canonical stack potentials should lie over. So they only actually increase it. I think I can see here. You can see. Okay. But they only really increase the stacking potential if for this space pair here, A plus T plus BTA. So this is actually plus the ABAT. This is the only increased stacking that I have. So I cannot say, okay, the stabilization only comes from stacking. It only shows in one single scenario. So I'm going to discuss this a little bit in a little. But from this parameterization, it is an oncogene probe design for Kira. So this is when we started our collaboration and everything. And her aim was to actually detect tumor DNA in the bloodstream. So you need very specific and very stable probes to detect this. And this is the reason she adds LNA. So her design for probes, I'm going to show this a little bit ahead. But for her design for probes is pretty much DNA to capture the target. And this DNA capture the target, this captured DNA capture the target. And then I have a linker DNA. So double sharing that this is an oncogenic sample. So if you bind here, the linker and the target and the capture, you're double sharing that the target is right. So she had a set. She wanted to predict the set for RAF, CRAS, and EGFR oncogenic probes. And we have a different probe for every single one of this, capture and linker. So what we did was, using our parameters, we wanted to predict the best site to insert an LNA modification. So we tested using the derived parameters, which I did this in my computer, if I can remember correctly. So you can predict this very fast. So I predicted almost 800,000 sequences for each probe, and for every single modification here, every single site, possible site of inserting a modification, and I predict the melting temperature here. And from this melting temperature, she chose the sequence that she thought was going to react better in her experimental set. So these were the sequences that she chose with these modifications. And after this, after choosing the sequences, and this is the difference, and this is the difference from the predicted, including the LNA, and without having any modification. For some, I have very different melting temperatures, so almost 10 degrees. But for some, I have very close. For example, this one here. So this grass 12 has almost the same. But when she predicted, actually, when we did the prediction and when she measured we had a correlation of less than 1 degree. So we could predict almost every sequence almost the same as she measured, as you can see here. But from sequence, we go a little bit different. We have a little bit different result. I'm going to discuss this in the opening. And to discuss this, why we did the same at the average openings for every single one of these probes. So, for example, here in blue, I have a breath capture that is not modified, and in red, I have the modification included. So you can see that where the modification is included is exactly where the regions prone to open before are inserted. So, for example, here I have a way less wider opening than for here in blue. And for AT, as expected, an AT internal, I'm going to have more wide opening. And if you can remember from the results that I showed from DNA, this wider opening is exactly the same result that we see at those high concentrations. So an AT at the terminal is going to be more susceptible to open than an internal AT. So, but LNA stabilized the sequences, the whole sequence. So, but if I have the breath linker, so this is an example of LNA stabilizing the whole sequence. So I have way a wider opening for the non-modified sequence. But when I put on the modification, I have a more closed opening. So this is going to now after the modified sequence, or non-modified sequence. Same way for cross capture, I have a stabilization when I put on the DNA. When I put on the LNA, the another cross capture, I also have a stabilization on the sides when I put on the LNA. The cross linker is also have this overall stabilization when I put on the LNA. Same way here, and same way here. So just to discuss this, this was really interesting because this paper was published. We assumed, actually, we did not assume. We discussed another paper that AT may have a third bond weight. This is assumed from 98, I guess, 1098. That AT could have a third weaker bond, so it's not actually a hydrogen bond. It's more than that van der Waals interaction, but it's still an interaction. And a few days before that we published a paper, this paper was released, and they actually proved it that AT has this possible hydrogen bonds here, and this could destabilize. So we actually, we cannot assume, but we pretty much, I don't know, infer that this third weak bond here could be strengthened due to the LNA modification for both adenine and thymine, especially because, let me go back, especially because we see almost the same potential for a canonical CG. So this could be doing 3 hydrogen bonds, and this weak bond could be strengthened. Sorry. And as the studies assumed, stacking could not account for the increased stability of LNA. So they almost have the same stacking interaction for a canonical. And in some cases, they even destabilize, for example, here. So they have lower canonical, they have lower potentials than the canonical. And interestingly, and confirming our results, this sequence here that has this increased stacking, this is actually the sequence that they measured. This is the only increase in the stacking that we have, is Lustate TAPAT. And that's the reason that they assumed stacking interaction as the reason for LNA stability. So a little bit of an outline. We applied the Bishop model to derive a complete parameterization of single element insertions, as we wanted to do in the first place. We see an increase in the Morse potentials, indicating stronger hydrogen bonding. Stacking potentials, they will depend on context, so it will depend which base pair that we're modifying. And the conclusion that we have here for the whole work, we developed an adaptation of the Bishop model to handle multiple strand concentration, increasing the scope of sequence that we can treat using the model. We modeled the terminal antilinophytes in RNA. We characterized single RNA modifications using the Bishop model. We did an oncogene probe prediction using this parameterization, so this could have actually more close to experimental situations. And as a future perspectives, I'm not showing everything that I did in the PhD here, but we were invited to write a book chapter for molecular structure of RNA by the owner of the software, so Vienna software, I think it's the pronounce, to our parameterization that we did using the nearest neighbor model. So this is done in variables, the software variables, for RNA at different cell concentrations. And we also drafting a paper using the nearest neighbor model for LNA, for single LNA modified probes. Although usually the Bishop model gives better results, people, experimentalists, they still prefer to use this nearest neighbor model. And I do not have, I lost all my data, but I didn't lose, but it's like stuck in my computer. But we are still drafting a paper with a different LNA modification. It's actually a peptide oncogene, not peptide oncogene, a peptide conjugate containing LNA. So this is one of the results. So I wanted just to show a little bit and to discuss this. And thank you all for listening to me, and my acknowledgements for CAPS. Any questions? I'm open.
Okay, good. Izabela, thank you for just let me change my settings here so that I can just a moment, please, so that I can see who is online. But I think Kira hasn't arrived yet. Before we start with the questions section, we are going for a short break, 5 minutes. Is that okay? So it's, we are back 10:20. Is that good?
Okay.
Yeah. So see you in a minute.
Eu passei
So we are
muitas
back.
horas do tempo. Vamos lá. Só tem nós dois.
Yes, you were a little bit over time, but I think that's not a problem. Right. So are we all back? Good. Okay, so thank you, Izabela, for the presentation. And we will now start with the questioning sections. And we were required to start with external members. And following academic tradition, we will start with the member who is farthest away, which I think no doubt is Luciana, who is sitting in front, right? I think Anderson agrees with that.
Absolutely.
Yeah, Rio and Belo Horizonte are very close compared to France. So Luciana, are you there? I think she's not back from the break yet.
Eu estou aqui.
Uh-huh.
Um minuto. Eu não consigo começar minha câmera.
Ah, é? Vai sem câmera mesmo.
You cannot start your video because the host has stopped it.
Did I?
Start my video? No.
It did?
I must have clicked on something wrong here.
No, it did start.
Agora, acho que agora começou, né?
Uh-huh.
Ok. Bom, gostaria de começar agradecendo, Gerald, pelo convite. É sempre bom estar em contato com você, seu grupo, meu antigo grupo. Obrigada. E eu sou super fã do nosso trabalho. Então, obrigado pelo convite. Bom dia para a banca, né? E eu, assim, particularmente, eu fico encantada com os trabalhos, gostei muito do trabalho que a Izabela veio realizando, eu acompanho a Isa desde pequena, né, Isa? E eu tenho algumas perguntas, eu vou passar um pouco longe da parte mais técnica com vocês, principalmente pelo seu primeiro trabalho, que foi um desenvolvimento metodológico, muito interessante. Eu tenho algumas perguntas, mas eu escolhi um caminho um pouco mais filosófico, a gente passando um pouquinho pela biologia da coisa, né? Apesar de bioinformata, eu tenho minha formação em biologia, então são algumas perguntas, vamos filosofar um pouco, assim. Eu quero aprender com você, assim, e ver a sua visão e entender melhor um pouco mais seu trabalho. Então, passando pela sua introdução, assim, tem acho que um ponto só que eu quero te fazer uma pergunta mais relacionada à escrita, mas não é nada demais, assim. Mas, por exemplo, antes, na sua introdução, você fala do trabalho com RNAs e altas quantidades salinas, menores quantidades salinas, todos esses experimentos que existem, os ensaios de NMR, etc. Você fala bem dos LNAs, que, assim, é muito legal isso. E você fala, assim, eu acho legal o contexto que você coloca, mas você fala muito bem de todo mundo, né? E eu chego lá no final da sua tese, e você fala pouco, né, das limitações da técnica. Eu estou na página 6.
Eu estou tentando
então,
achar
assim,
a página.
Um bate-volta. Então, eu vou na sua introdução, você fala super bem de todo mundo. E lá no final você fala um pouquinho de uma limitação do RNA que é financeira mesmo, né? Mas, assim, esse mundo é um mundo um pouco da fantasia, eu queria um pouco mais um mundo real, assim. Dentro do que você tem visto e do seu trabalho, nos dois papers que você apresenta, quais são as limitações das técnicas que você tem visto? O que, claro, além do que você melhorou em termos de cálculo, etc., o que você vê no contexto de limitação de técnica?
Eu acho
Do
que,
que existe já?
no geral, o que eu mais vi foram limitações em termos de experimentais mesmo. Porque, por exemplo, quando falando de RNA, por exemplo, você vai sintetizar um RNA, você precisa checar se aquilo ali vai formar uma estrutura secundária. Então, você tem que checar se aquilo vai ser duplexo mesmo. E aí você vai ter RNA no laboratório, que você vai ter problemas, por exemplo, para sintetizar, porque ele degrada fácil. O sintetizador é diferente, não, acho que é o mesmo, né? Onde eu lembro. Mas eu acho que parte muito mais desse pressuposto de síntese. E aí, quando você faz a síntese, beleza, você conseguiu sintetizar, fez tudo bonitinho, você ainda tem problemas de você conseguir entregar isso celularmente. Você conseguir fazer essa entrega disso, porque grande parte, principalmente do trabalho com a RNA, é você tentar fazer a entrega biológica disso, né? Então, esse problema de entrega, de estabilidade, de você conseguir passar o núcleo, conseguir passar a membrana celular, por tudo que eu li, aparentam ser grandes problemas para esse tipo de coisa. Então, assim, apesar de ter muito potencial, coisas com RNA e RNA para efeitos de tratamento, você ainda tem muitas limitações nesse sentido. Assim, até onde eu sei. Sei que existam outras, mas até onde eu li era isso que passava, assim, sabe, de cara.
Você vê que é um problema metodológico, experimental.
Sim, eu acho que tem um problema muito experimental. Então, por mais que a gente prediga, por exemplo, a melhor sonda possível, pode ser que quando você faz isso in vitro funcione, e fazendo todas as condições que você previu, e quando você faz in vivo não funciona, porque às vezes é uma interação que você não predisse, uma interação que você não conseguia, assim, saber de princípio, ou então você não conseguiu entregar na dosagem necessária. Então, os grandes problemas de sondas modificadas com LNA, principalmente, são esses. E toxicidade também, é claro, né? LNA, acho que teve uma droga aprovada, que é acho que eu desculpe isso, o Formivizen, mas as últimas coisas que eu li fala que ele foi descontinuado, porque ele tinha uma toxicidade no fígado bem alta. Porque você tem que colocar bastante coisa para você conseguir ter eficácia, né? Você tem que dar doses muito altas.
Ok, eu estou alternando entre o microfone do meu computador e o meu fone de ouvido aqui para eu conseguir te escutar melhor, que tá bem baixinho para mim aqui. Por exemplo, dentro do seu trabalho, né, que antes só para falar de um pontinho, que alguma coisa que eu vi aqui nos seus objetivos, você só, não sei se é uma correção ou se eu entendi mal, porque pelo que eu vi, o seu primeiro trabalho, o seu primeiro paper, ele fala da aplicação do Payer-Bishop Model para sequências com different strand concentrations e e concentração de sal também. Por exemplo, aqui você coloca obtain a parameterization of the PB model for sequence at different strand concentrations and salt concentrations as well, ou é só...
as well.
Porque embaixo no específico você fala concentração,
dois, né?
strand concentration and salt concentration.
Isso, não, eram os dois, você está certo. Eu deveria ter colocado, talvez para ficar, né, coerente com o restante, multiple strand concentration e salt concentration.
Ok, é só um pontinho que eu vi, assim, que ficou um pouco para mim
confuso.
Claro, mas eu falei, eu vou tirar a dúvida com ela. E Bom, assim, de uma forma geral, sua introdução, o que você falou sobre de uma forma geral, né? O que que tem sobre as sondas, o que que tem sobre os trabalhos de sal, de concentração de sal e as diferentes metodologias que existem de LNA também, eu achei muito legal. Talvez você discutir um pouquinho mais as limitações. Eu gosto muito de ver o paradoxo, sabe? Quando você tem coisas que são muito legais e depois você fala, olha, mas tem essa limitação, isso precisa melhorar nesses aspectos X e Y. Assim, eu acho interessante, porque sempre tem limitação, né? A gente trabalha dentro de um campo muito limitado, e quando você passa para... E nem se fala, porque a gente trabalha dentro de modelos que são compartimentados, né, de laboratório, e isso não mimetiza a fisiologia celular, né, que eu acho que esse é um dos pontos principais, assim, de técnicas desenvolvidas para diagnóstico, e depois se você tenta adaptar num contexto em vivo, é super complicado. Então, eu acho legal transitar dentro desses dois mundos, sabe? Olha, isso aqui é muito bom, mas, olha...
Mas eu tenho esses problemas.
Isso. E passando, por exemplo...
A impressão que eu tenho é muito que, assim, desculpa, a impressão que eu tenho muitas vezes é que os trabalhos, eles discutem muito o lado positivo. Ah, isso é positivo para isso, isso e isso. E aí, quando você vai ver trabalhos que tentaram usar, aí você tem, tipo, deu errado nisso. Tal coisa não é eficaz por causa disso. Então, às vezes, falta mostrar mesmo os dois lados da moeda. Desculpa.
Estou de acordo com você. Tem 2 papers da Nature que fala que a gente devia publicar dados que não deram certo.
Sim,
Metodologias que não deram certo, e você vai impedir que outras pessoas repitam o que você fez, assim, você ajuda, você ganha tempo. Mas a gente ainda precisa caminhar para esse lado da moeda também, né? E, por exemplo, no seu primeiro paper, assim, eu achei muito legal o que você fez, que você adaptou a técnica para analisar dados de RNA com diferentes concentrações salinas, né? E, por exemplo, vendo esse seu trabalho, né, que você pega, porque, claro, você falou o tempo inteiro você fala, né, de problemas de base de dados, porque você tem base de dados heterogênea, né, você usou base de dados públicos, você tem base de dados heterogênea, e esse é um dos grandes problemas que a gente enfrenta, assim, para todos. Você não é a felizarda, a única felizarda, isso está de uma forma geral. Hoje, com as análises multiômicas, por exemplo, e integração de dados, que hoje o que está acontecendo é a integração de dado clínico com dados ômicos, e existem muitos dados públicos e dados que são privados. E para você fazer a integração desses dados e conseguir harmonizar os dados, que a gente passa por um processo de harmonização, é extremamente complicado. Você tem que usar modelos estatísticos, porque o grande problema desses dados, ele passa por como é que eu falo isso? Ele passa por valores extremos, porque quando você tem dados quantitativos vindo de diferentes experimentos, né, você tem normalmente os resultados de base, mas você encontra valores extremos. E tendo diferentes abordagens experimentais, é um grande problema você harmonizar esses dados e conseguir fazer uma análise comparativa entre eles.
Porque às vezes tem dado, mas não tem estatística.
Exatamente. E é complicado você inferir alguma coisa quando você tem valores extremos, por exemplo, média, essas coisas não respondem muito a questão. A gente usa modelos de regressão logística, mas ainda assim
é complicado ainda em termos, né?
É uma limitação. E eu fiquei curiosa para saber, porque eu vi que você adaptou vários parâmetros, você fez a conta aqui, que foi assim, eu olhei e falei assim, eu não vou nem tentar entender essas contas. Mas eu fiquei curiosa para saber dado que você fala o tempo inteiro, né? A gente não tem... Você tem dados vindo de diferentes condições experimentais, diferentes concentrações salinas, e eu fiquei curiosa para saber como é que você fez, se você fez alguma harmonização para que você conseguisse pegar, assim, um mínimo de dados homogêneos para fazer suas análises. Como é que você fez isso?
No RNA ou no LNA?
No seu primeiro...
Você ficou muda, Lu.
Salt Dependent Mesoscopy Model for RNA at Multiple Strand Concentrations.
Claro.
É o seu primeiro paper da sua tese.
Na verdade, quando a gente faz esse tipo de análise, o que a gente tenta, e o que eu tentei também no LNA, é tentar manter um conjunto mais homogêneo possível nesses termos de diferenças. Então, por exemplo, o LNA era quase sempre o mesmo grupo que media, nas mesmas condições, e tendo mais ou menos a mesma coisa, justamente para você evitar essas flutuações. No caso do RNA, Gerald me corrija se eu estiver errado, foi um caso, assim, diferente, um pouquinho diferente, por quê? A gente tinha esses dados de RNA, eles funcionavam muito bem no VAR-BIS, só que para o Perarbishop não tratava, então é quase tudo de uma coisa só. Tanto que a gente discute, eu até esqueci de discutir isso na apresentação, mas os dados que a gente tem que a gente faz essa correlação, essa correção para, não correção, né, adaptação para múltipla concentração de fita, eles vêm de uma base só, eles vêm de um cara só, e foram dados que eu tratei antes, então eu limpei quase tudo que é erro antes de fazer, e aí a gente tenta conjugar isso com dados de 98, que é do XIA 98. E aí quando a gente tentou fazer colocar, eu briguei muito com o Gerald, eu ficava, Gerald, os dois não casam, um é um trem, outro é outro, não faz sentido. Ele falou assim, não, mas não tem como você não discutir. Então, assim, a gente até discute no paper que a comparação que a gente faz entre os dados do Shen, que é de 71 a 621, não pode ser diretamente relacionada com 1021, porque 1021 são condições completamente diferentes. Grupo diferente.
Vocês trataram de forma... Então, vocês trataram esses dois datasets separadamente?
Isso, aí o 1021, aí eu esqueci, desculpem, inclusive, eu esqueci de mencionar isso, porque a gente tinha que achar um F que fosse não bom o suficiente, mas um F que desse bons resultados, justamente porque a gente queria comparar com os resultados do 1021, que o Gerald já tinha minimizado antes, que é aquele RNA em concentração mais alta, e a gente tinha que colocar isso no paper de alguma forma, então a gente tinha que tentar correlacionar o que a gente obteve, como que isso varia com concentração salina e o 1021. Então, sempre era um problema. E o 1021, o que eu faço com isso? Porque era um monte de sequência completamente diferente. Mas, assim, ainda assim, a gente vê que é parecida a variação, apesar de ser um pouquinho brusca ali no início. Se eu voltar aqui, tá vendo? Mas isso foi uma coisa assim, eu briguei muito com esses dados.
Eu imagino.
Briguei muito. Eu queria matar o cérebro. Aqui, ó, nas regressões, eu esqueci de mencionar isso, acho que eu já fiz isso tantas vezes. Nas regressões, vocês veem a tela ainda, né? A gente tem aqui 4 valores, né? Quando eu faço com Fs diferentes. Mas aí, quando eu comparo mesmo, que é F igual a 1, eu comparo com o 1021, que é essa minha quinta aqui, ó. Minha quinta linha de pontos, né? A quinta coluna de pontos. Tá vendo? E aí, apesar da gente ter essa diferençazinha aqui, eles ainda seguem bem parecidos com a linha aqui. Mas a gente
então,
não faz a comparação quantitativa. Pode falar disso.
Não entendi. O seu 1021...
Não dá para disputar.
Foi o controle que você usou? É um tipo de controle que você usa?
Na verdade, não chegou a ser um conjunto controle. Eu não posso chamar de descontrole, porque a gente minimizou também. A gente considerou terminal para ele. O Gerald já tinha feito essa minimização normal, sem considerar terminal, sem considerar nada. Acho que é um dos primeiros trabalhos do Gerald. Primeiros não, né? Foram as confirmações do Ferreira, do VARPAR em si. E aí a gente tinha mais ou menos assim, o que a gente esperava obter. deveria ser parecido. Então, a gente tentava comparar muito com o DNA. Os resultados que a gente já tinha para DNA, que são aqueles comparando todos. Aí, quando a gente fala, por exemplo, de condição experimental, os dados do DNA, eles são medidos por um grupo que ele faz patente. Então, é um grupo que é muito mais metódico nas medições. Então, são dados que são muito melhores. Eles não têm tanto problema igual a gente teve com esses.
Entendi. Então, sua referência, né?
A gente já tenta mais ou menos saber, assim.
E, por exemplo, ainda nesse contexto, assim, de dados, né? Por exemplo, você sempre fala, né, várias vezes dessa dificuldade dos dados. A gente falou da dificuldade de harmonizar os dados, que é um problema geral, assim. E, por exemplo, ao que especificamente para dados de contendo temperatura de melting, dentro do que você tem visto, assim, o que você atribui essa dificuldade? O que você acha do problema de você ter poucos dados associados à temperatura de melting e poucos dados associados a essas diferenças para analisar ácidos nucleicos imersos dentro de grandes concentrações salinas, baixas concentrações. Você tem alguma ideia por que isso acontece? Porque, por exemplo, o primeiro trabalho Santa Lúcia, acho que, se eu não me engano, tem mais de 20 anos que começa esse trabalho, e ainda assim, por que você acha que a comunidade não...
eu entendo.
Difícil acessar esses dados?
Assim, o padrão que eu vejo, grande parte desses dados que a gente extrai e parametriza todos os nossos modelos, em geral, são parâmetros que foram derivados tentando calcular próximos vizinhos, tentando calcular parâmetros de próximos vizinhos. Então, o pessoal que já estava tentando fazer um modelo. Então, por exemplo, quem está tentando medir só, por exemplo, o carinha que mediu para o stacking, e falou que o stacking é o que mais estabiliza a DNA, ele mediu uma sequência, o interesse dele era aquela sequência. Mas aí, quando eu falo de um modelo, eu preciso de todas as possibilidades. Então, tipo assim, eu preciso de ter variações, eu preciso ter de mais de uma vez essa variação. E isso só é feito no próximo vizinho, geralmente. Então, tem muito pouco dado. E aí, às vezes, esses dados... Ai, perdão. Dá para me escutar? E aí, às vezes, esses dados, eles vão para a patente. Então, por exemplo, os dados do O'Giscali, eu acho que ele foi muito gentil em publicar, porque quase todos eles estão patenteados e são do IDT, que é o pessoal que como é que chama? Que vende sonda. Então, eles sintetizam sonda para vender. Então, assim, tem muitos grupos que nem fazem isso. Tem grupos que vendem sondas e não publicam dado, porque para eles é desvantajoso, não faz sentido, é chato de tratar. Tanto que esse artigo com RNA foi uma colaboração que o Jared arrumou com o Brent, e ele é do Chain 13, que é o que a gente fez, alguma coisa que eu não lembro. Na verdade, ele extrai dados de próximos vizinhos para RNA em concentrações diferentes, em concentrações salinas diferentes. Ele extrai justamente isso, e aí a gente pediu o dado cru, que é esse dado com várias concentrações de fita, porque a gente queria tentar tratar um maior volume de dados, porque tinha muito pouco no artigo. Quando esse dado chegou, era uma bagunça. O cara parece que ele colocou sequência no Excel, sabe? Então, assim, tem toda essa dificuldade. O cara não está acostumado a lidar com dados, o cara está no laboratório sintetizando. Então, assim, não chega arquivo que você consegue falar assim, dá para tratar. Entendeu? Às vezes a gente tem que passar por muita raiva.
Sim, e, por exemplo, em termos de interesse da comunidade científica para trabalhar nesse tipo de modelo e melhorar esses aspectos, o que você acha, assim? Porque, por exemplo, durante anos a gente usou modelos empíricos, né, cálculos empíricos para modelos empíricos para calcular temperatura de melting. E, assim, já vi muita gente desenhando sonda na mão mesmo, e fazendo aquele cálculo de energia livre na mão, e, bom, funciona, né? Não posso dizer que não funciona, eu mesmo já fiz muito. E, por exemplo, com o modelo que você trabalha, né, claro, você tem mais do que temperatura de melting, você pode medir a flexibilidade, você vai ver o stacking entre as bases, você vai ver a interação também de hidrogênio entre as bases, de uma base, duas bases. Isso é um modelo muito mais
parâmetro físico, né? Eu não estou falando de parâmetro empírico, por exemplo.
Exatamente, é um modelo muito mais refinado. Mas, por exemplo, para uma aplicação técnica, para você falar assim, não desenhe primer na mão, use o meu modelo. Como é que você falaria isso? Para o pessoal do laboratório da bancada.
Sim, porque assim, você joga essa contaiada toda que a gente faz, eu acho que você teve um pouco no modelo, você joga isso pro cara que está sintetizando, ele fica, não, vou fazer lá, não, porque o próprio IDP já dá um negocinho para predizer, né? Por isso que eles fazem. Você seleciona uma sonda estelar, eu preciso que isso denature a 60 graus Celsius. Aí você faz um cálculo aproximado, é isso mesmo. Mas, por exemplo, o LNA, a Kira estava tentando fazer sondas assim, e às vezes ela tinha umas que falhavam assim, absurdamente. Pelo modelo, você supõe que é estável, mas quando você vai ver, o negócio falha completamente em vivo, a temperatura é muito nem vivo. Você vai fazer a temperatura de melting e já está completamente diferente do que você prediz. Então, às vezes, o nosso modelo pode ajudar a entender por que dá tão diferente. Que foi justamente uma das coisas que a gente achou no LNA. Nem sempre você colocar o LNA vai estabilizar. Então, tem situações que você coloca o LNA que vai completamente mexer com a estabilidade da sonda e você vai ter um negócio completamente diferente. Eu acho que isso faz muita diferença. Então, por exemplo, uma coisa aqui que eu tenho que chamar muita vez a gente conseguir fazer, acho que já era de tempo há uns anos, é a gente conseguir predizer estrutura secundária de RNA. O método hoje para fazer isso é completamente empírico. Então, às vezes, o cara ele chuta a sequência de RNA e fala assim, ó, tomara que não vire, sei lá, um bulge. Tomara que não vire um hairpin. Tomara que... Entende? Acho que a gente conseguiria dar mais termos físicos para falar por que não foi, por que não está dando, onde eu faço, o que dá para melhorar. Eu acho que é nesse sentido.
Então, por exemplo, eu tenho uma outra pergunta para você. Nesse contexto, né, que vocês podem ajudar a melhorar e ajudar as pessoas, porque eu acredito muito nisso, principalmente no que eu vi do trabalho de LNA, que eu vi exatamente o que você falou. Como dar acesso à comunidade científica para usar os modelos que vocês produzem de uma forma fácil, que esse é o challenge, esse é o bottleneck, né, como que eu ponho, porque, por exemplo, quem desenha sonda, né, não é, assim, hoje o Bioinformata faz esse trabalho, eu já fiz muito, assim, usando ferramentas, né, computacionais, mas como que, porque o cara da bancada, o biólogo da bancada, ele não quer rodar linha de comando e usar algoritmos e programas super complexos para obter isso. Então, por exemplo, vocês têm algum plano, alguma ideia para poder tornar isso acessível para o público da comunidade científica de uma forma, assim, fácil, né? Que seja sedutor. Olha, vem aqui, aperta esse botão e você vai ter o que você precisa.
O Gerald me corrija também, mas o Gerald, ele mantém um website para predição de predição de temperatura. Só que essa predição de temperatura usa o modelo próximos vizinhos, que é o que o pessoal conhece, né? Porque, por exemplo, se quisesse usar o modelo próximos vizinhos na linha de comando, existe o VARDIX, e para o modelo Operar Bishop existe o TFREG. Então, mas aí você vai falar com o cara, olha, você tem que rodar esse script e você tem que saber entender os resultados. Eu acho, lendo o manual do TFREG, eu acho que dá para entender. Se você tem background nisso. Se você não tem, o cara fica meio perdido. Então, eu acho que talvez faltaria uma ferramenta parecida com a do IDP, que é provar, não para o VARDIX, porque já tem, mas para o TFREG em si, para rodar isso e conseguir obter as coisas usando o modelo Operar Bishop, e falar, olha, aqui é melhor. Mas para obter parâmetro físico, para a gente olhar e falar assim, Por que isso aqui não estabilizou, por exemplo, no caso do LNA? Aí a gente precisa de uma análise maior, a gente precisa parametrizar, a gente precisa olhar gráfico, precisa visualizar o problema. Agora, só para dizer temperatura, talvez uma estrutura parecida com o TPM cálculo, que o Gerald tem.
Ok. Fica a sugestão e a pergunta aí para os próximos trabalhos, né?
Deixa eu só fazer um comentário e... Ô, Lúcio, está me escutando? Tá, deixa eu só fazer um comentário em cima disso, porque isso não é exatamente o assunto da tese da Isa, né? Mas como não sei quantas pessoas estão assistindo no YouTube, deve ser metade do planeta, mas a sua pergunta, ela realmente aborda uma dificuldade que a gente tem, porque a gente ainda está num processo de validação do método, né? Então, muito também do que a Izabela fez agora e outros trabalhos, né, o seu, inclusive, quando você esteve no grupo, era de validar, mostrar, olha, o método funciona, ele dá resultado consistente, o que a gente obtém não é maluquice, e agora a gente embarcou numa fase em que a gente busca parcerias experimentais, né? E aí entra a Kira, né? Com a parte dela, para trabalhar diretamente junto aos grupos experimentais, porque a gente tem essa consciência que você acabou de falar aí. Eu não vou... é, é, eu não tenho ilusão que as pessoas vão pegar os manuais dos nossos softwares, embora eles sejam todos públicos, né? Para rodar isso em linha de comando, nem mesmo em interface web, porque essa parte do Perabishop, ela não está pronta para uso dessa maneira, né? Assim, se você não entende absolutamente nada do Perabishop, não adianta uma interface web, porque não é por aí que vai. Então, o que eu penso que a gente vai acabar vendendo, né? Se é que isso pode ser vendido, é a força de trabalho que a gente forma de pessoas que conhecem o modelo, sabem implementá-lo, sabem usá-lo e produzir aplicações, né? Como essa aplicação que a Izabela produziu de fazer uma coisa que não é possível fazer de outra maneira, que é simplesmente testar todas as modificações do LNA, uma coisa meio ignorante, assim, né? Vamos testar todos e ver o que dá. Que é o que o modelo permite fazer, porque ele acaba, pela simplicidade, ele é de alto desempenho, né? Então dá para fazer esse tipo de coisa. Mas eu acho que é isso, eu acho que a interface nossa são as pessoas que a gente está formando e que ou conhecem o modelo, ou sabem explicar, ou sabem implementar, né? Pelo menos eu penso um pouco nessa linha.
Talvez chegar no nível.
Sim, mas eu acho que é bem por aí. E eu estou de acordo. Mas é só uma sondagem, assim, para ver como é que isso vai caminhar, qual que é a direção, as direções que isso caminha. Mas, assim, obrigada, Gerald. E, Isa, passando para o seu segundo paper, né?
Uhum.
Então, eu fiquei assim, é por isso que eu pergunto como é que é essa coisa de espalhar para a comunidade científica de uma forma fácil também, que eu sei que entendo que isso é difícil, mas eu sempre fico empolgada quando eu leio esses trabalhos. E, por exemplo, eu não sei se eu entendi muito bem, mas pode ser uma pergunta boba, mas você usou alguma coisa do que você melhorou no seu primeiro paper aqui, da dependência salina, né? Para isso que você usou para análise de LNA?
Não diretamente. Essa análise que a gente fez para a Kira, como eu discuti, né? Eu acho que faltou isso também, às vezes, na discussão da apresentação. a parametrização que a gente fez foi em concentração alta de sal. Então, a gente teve que colocar uma correção salina. É uma correção salina proposta pelo próprio History, ele propõe, acho que no 04, para DNA. Como a gente está falando de análogo de DNA, acabou que essa parte do RNA não entrou muito. Eu cheguei a testar uma correção salina de RNA nessas sequências, mas a de DNA performa melhor, em termos de predição. E mesmo usando a correção, a gente cai muito em cima. Assim, acho que os parâmetros ainda são bons o suficiente para ter conseguir lidar com essa variação.
E você, porque eu vi que você usa sódio nessas análises, né?
Sim.
Você teve a oportunidade de olhar magnésio? Por que não magnésio? Porque
A Kira chegou a... Desculpa, era só isso? Praticamente. Travou? Não, né? A Kira ela chegou no dia 1 antes de sequência com magnésio. Quando eu fui pro sanduíche, a ideia era eu sintetizar algumas ondas e fazer um melting com solução de magnésio. Só que pandemia. E aí, quem está fazendo a parametrização, se eu não me engano, é a Izabela, do grupo, com magnésio. Aí, considerando esse LNA com magnésio, porque ela já tinha feito um trabalho com magnésio. Deu para entender?
Just to let you know that Kira has arrived.
Ah, ok. So,
Isabel, estou em magnesium, but in canonical DNA. We don't have the data for magnesium and LNAs as far as
I think Kira has some sequences with LNA and also measure in magnesium, but they're not enough to do a minimization to actually parameterize. So that's the reason.
Luciana?
Você tá muda, né?
We don't hear you.
Hello?
Hello?
Can you hear me?
Yep.
Yes.
Okay, sorry about it. I don't know what happened. And for example, I wrote your methodology, and I'm still in doubt. For example, how do you construct if you use it, control data set, and if yes, how did you construct it?
No, we did not use a control data set for any of the parameterizations. Actually, for the work that I'm doing now, we are considering a validation data set. And also for RNA, we have a small validation data set, but it's not, it cannot be called a control.
Okay. And how do you compare, for example, if you don't have control data set, okay, and for example, you need to compare, for example, the Watson-Crick sequence, for example, without modification, right? Against your sequence with LNA modified,
Okay.
right?
Okay.
And which kind of, do you consider it the salt concentration for both data set, for your LNA and your DNA unmodified data set?
Okay, okay, I got your question. Actually, we had parameterization for DNA at high salt concentration. So that we use it to extract, I think you can see my screen, you can see my screen. But from this, we use it to extract the data, actually to restart the parameterization for LNA. So we consider that LNA is going to behave the same way as canonical DNA, and we let it optimize. So this is going to change. So that's the reason LNA has different potentials. And then we use this canonical DNA, and we also let it optimize with the model. So it not have a much change in the canonical base pairs, in the canonical parameters. And the data set also has sequences without any modification. Actually, the analog sequence. So if I have a sequence with a plus A, this sometimes also have the canonical sequence. Make sense?
Yes, okay. And for example, I'm just wondering regarding the capture linker probe design that you did.
Okay.
And you mentioned melt temperature above 45 Celsius degree, right? And what is the size, the length of your probe, the average?
Actually, when Kira gave me the targets that she had, she said, okay, I need 22 nucleotides per sequence, and I need this to achieve at least 55 degrees. So the canonical sequence has to, if I understood correctly, has to achieve at least 55, and the modified one has to have an increased melting temperature from 55. So that's the reason she chose these probes. I think it's the way the annealing system that she does.
Okay, this is go out from, okay. And for example, do you have, I didn't see it on your paper, even in your analysis, how can you compare, for example, how can I say it, current technicals that we already use for detect mutations and specific markets, and how do you measure the difference, how can I say it, the performance between the analysis that you did, for example, and the correct technicals that already exist, which is different for LNA, for example. Do you have, for example, data or some analysis regarding the FDR, false discovery rates, to see the rates of your false positive data? Do you have something to measure it? Do you did something?
Okay, so you mean at the efficiency of the probes. If the probes are actually efficient in the in vitro environment. That's the question?
Yes, my question is regarding, for example, when you have this kind of technician, what is the false positive rates you
So if
have?
do you mean by false positive? If I have a good probe, so this is going to bind in the target with successfully, and I have a bad probe, so I predict this is going to be a good probe and it's not binding. That's the question.
I can't hear you very well.
I'm sorry.
Very bad. The audio. Can you try to? Pode tentar chegar mais perto. Tô com dificuldade de te ouvir, Isa.
Wait. Let me just get a little bit. Excuse me. I'm using the audio from my phone, so that's the reason. So Zoom is not really happy with my microphone from the computer, so that's the reason. But it's improved or not at all. I can also try to speak louder.
I will try to change my question. When you do a kind of analysis, for example, your algorithm, you give some results. Normally, most of them are true, really true. It means you have good results, but you can find mixed with your results that you feel that is true based on scores, value, etc.
Yes.
But it's not really true. You know,
yes, I think it's what I thought. It's just like you're saying, if I have a good probe prediction, so if this probe is going to work. So for this question, we are not there yet, because I think Kira can update us on this, but I think one of her students are actually doing the binding and the hybridization with the oncogene targets. So we just did the probe prediction in terms of melting temperature. So if I want to compare melting temperature prediction, because that's what we did at the end, we can compare with nearest neighbor model. So the predictions that, for example, IDT gives, but we cannot compare in terms of probe efficiency or probe binding, not yet.
Okay. Okay, very nice. So I think I finished my questions for you, for your thesis. I would like to say congratulations for your work. It's really nice. It was very nice to read your work and read what you did in terms of science. You gave a very nice contribution, and I'm sure you how can I say, you shine the next step. So can you share with me what would be your next step? What are you going to do?
Actually, I think with Gerald, I'm going to finish the 2 papers that we are drafting. But at the job that I got is a little bit different, because they use single cell data to predict cell interaction. So it's a little bit different that I did until now. But with Gerald, I have the 2 papers still to finish.
Okay, very nice. Thank you very much.
Thank you. Thank you very much for accepting.
Okay, Luciano, thank you very much for your comments and questions. We now move on, but before that, we would just like to say hello to Kira, who has just arrived a few minutes ago. Kira, whenever you like to make any comments during the questions, just feel free to open the microphone and speak up.
Yeah, thanks. So it's a pleasure, and I apologize, I'm a little delayed. Yes, I do have some comments and questions, but maybe I should wait till all the sensors are done. Okay, should we do that?
Right.
Thanks. It's good to.
So Nice to see you. So we move on to Anderson, who is the other member, external member from the Federal University of Rio de Janeiro. Anderson, are you there?
Yes.
Okay.
Well, so I'd like to start thanking Gerald and Izabela for the invitation to read and examine your thesis. I was very impressed, positively impressed by your writing. I think you did a great job. Not only the writing, but obviously for the content of the thesis and how dense and deep your work is. But I was very impressed by how concise the thesis is, and I think this is great. And I really want to commend you on that. It's very easy for a scientist to just, write about the things that we want to convince people that it's interesting. But it's very difficult to use few words to say the important things, and I think you did that very well. So I really want to commend you on writing a good and concise text, which is not difficult, which is very difficult. It's not easy, and it shows a lot of growth. I think, yeah, for a student to do that. I also want to commend you on the fact that you wrote in English, which is also not easy for most people, and I'm sure that this international experience you had was very important for you, and this is really the way to go. And also to defending on English, which is also not an easy thing to do also. So to be able to communicate and to defend your thoughts and your ideas in another language it's really not easy. So I really want to commend you on all those things. It was a pleasure. It was a pleasure to read your thesis. I'm not, obviously some parts are a little harsh. As a non-physicist, it's not easy to go through everything and all the equations and stuff, but I think I could understand the big picture, the whole idea, and you also made yourself very clear during the presentation, so it really helped me understand some things that I couldn't understand by reading the text. So some things got more clear when I actually saw your presentation. So I think I could grasp the whole idea, and I think you did a wonderful job. And we're going to talk about some things. We're going to discuss like doubts I had, but none of that is going to take away the beauty of your work. It's extremely well done. It's extremely well conducted, and I would like to comment on that. All right.
I'm glad that you like it.
So now, Izabela, I'm gonna go through like page and page. I'm a little bit more okay. So just a few things. Some sentences, some things that was not absolutely clear to me, and we can discuss, and then we move on to your results. So on page 19, let me see. Yeah, there's one sentence that I found it a little bit, when I read, it was like, what exactly is she trying to say? And maybe you can clarify it to me. So you're talking about you're on the second paragraph, you're talking about the effect of temperature and salt concentration on the structure of double-stranded RNA. But then you use it as... Paper. I'm sorry?
You mean in the paper, right?
No, I mean in page 19 of your thesis, of your document, in the introduction.
The pagination is different, I'm sorry.
Oh, okay, I'm sorry. Oh, I'm sorry, it's page 3, right? Yeah, well, yeah, I think it's page 3. Yeah, it's page 3, sorry, my bad. It's under 1.2.1, silent effects in double-stranded RNA.
Okay, I'm sorry about the comment.
No problem. So in your second paragraph, you're talking about the stabilization of double-stranded RNA and how the structure of RNA changes with physical-chemical parameters such as temperature and ionic strength and stuff like that. But then you use it as an example. Use it as an example of the binding of a double-stranded RNA to a protein, right? And how changing salt concentration changed the binding. I thought was a little was a little odd because, I mean, if you're changing the ionic strength of the environment, you're probably changing the electrostatic contribution of the interaction between the RNA and the protein. And you may or maybe not changing the RNA structure itself. Do you know what I mean? So as
Yes.
you're trying to introduce the effect of these things on the structure of the RNA, I'm not sure if this is the best example to give.
Okay.
Do you know what I mean?
I know the problem. I know exactly the problem. The sentence is better clarified in the paper because this interaction, this protein interaction, is this PKR. Is that the protein? And depending on the salt concentration, there's a little change of conformation. It turns out to, how can I say, to impact the binding. So the binding is not so well, you know? So it's not so well binded when you change the salt concentration. I just didn't put the example here, the full example here, because then it will be too close to the paper. But that's the protein that I was saying.
Yeah, I mean, I think it's hard to differentiate between what's going on between the RNA and the protein and what's going on with the RNA itself. I mean, it's an example. It's just the way it's stated, right? Let me see. Another thing that maybe you can clarify to me within this paragraph, in the end of this paragraph. So this final sentence says you're talking about differences between the double-stranded helices of RNA and DNA, and how they differ in terms of geometry and things like that. And it's you said something that I didn't really understand. So that these differences may be responsible for a different condensation behavior or multivalent ions. And I was just like, where is she going on?
Yes.
So I really didn't get this part. So what is exactly this?
I remember the paper. I also remember the paper that states this because they measured this electronic binding. So they have this whole electronic surface. So they can measure where the ions they are going to bind. So yes. And they measure the both surfaces for DNA and for RNA. And they have different, actually, it's not very, very different, but it's not slightly different. So they have a little bit different from each other in terms of electrostatic potential. So where the ion is more prone to bind. Does make sense? So that's the reason of this sentence.
Yeah, I think what I didn't really understand was the term condensation. You know, like a different condensation behavior or multivalent ions. I had a hard time.
It's ion condensation.
Yes,
so
exactly.
It's going to condensate. Okay. So it missed this part of the ion condensation.
Yeah.
Because paper is exactly this. They measured this different electronic surface, and then I don't remember if the same paper, and then this directly correlated with different flexibilities of both helices. Does it make sense?
Yeah, a little. So let's move on. Well, something else is still in the introduction section that maybe you can comment to me.
Yes.
I was like, I mean, that first of all, I didn't know, and I was somewhat impressed by this. But so you have this sentence that says that when you have a purine position in the 5' end, that it changes the stability of the 3' end of the RNA, right?
Yes.
So and there's something to do with mismatches, and it was not really clear to me. Could you comment on that?
Yes, yes, of course.
I mean, it's a little bit.
Odd.
Yes, you wouldn't imagine that a substitution in one end of the sequence would make such a difference on the other end of it. So, I mean, what is the mechanism?
Yes, I think is the but yeah, but I do remember this. Yeah.
Because here you're talking about base frame, right? So the fact that
yes. Actually, I think we can almost see the same in the test figure here. But Cheryl didn't want to discuss this in the paper. But can you see that this? You can still see my screen, I guess. But this end is opening more than this end. So it doesn't seem to depend on which base pair that you put on the 5 prime. 5 prime is still going to be more prone to open than the 3 prime.
I think it's Izabela. I think it's the way the phrase is written. It gives the wrong impression.
Oh, okay.
I think what you wanted to say is that the purine, if you put it at the 5 prime end, one thing happens, but if you put it at the 3 prime end, something else happens.
Yeah.
And the way it is written, it gives the impression that the purine at the 5 prime
causes
influences the other side of the word, right?
Yeah, and I was like, wow,
no,
there's got to be some interaction between these two tones. Okay.
No, it's the way it was.
And the way it's stated, yes.
Thank you. I think it's the way we formulated the sentence. So it gave this wrong impression. I'm sorry. I will correct this again.
No problem. Okay.
So just let me mark.
So talking about base frame, just also about the writing. In this sentence, it's base frame is an important aspect of RNA stability. In particular, it's unclear how frame depends on salt concentration. Maybe you could also add that it's unclear how base frame is dependent on nucleotide sequence too, right? Because it's something that you're actually trying to tackle with your work. So just to make it a little bit complete of the idea. So what are the other things that may influence this specific aspect of the structure of DNA?
Go to the question again. So your question is
no, it's not a question, it's a suggestion in writing that you add to this sentence that base frame is dependent. You're trying to understand how base frame changes with the cell concentration, but also with changes in nucleotide sequence, how it depends on the sequence. Because I think that's very interesting.
Okay, yes, I will discuss this on this sequence and try to discuss the discussion.
Okay. Also, Izabela, on the 4th paragraph, the first sentence, you said, more recently, we had access to a data set of measured temperatures of the sRNA at multiple strands. I would say at multiple strand concentrations.
Okay.
Then, otherwise, it gives you an idea that you're going to have, like, I don't know, triple helix or something, quadruplex, all the strands, right? But it's multiple strand concentrations then. And
yes, you're right.
Experiment was done at different concentrations. So.
Yes, I will correct this.
Let me see. Okay. Yeah, well, another sentence that I had, I'm sorry, I'm very writing oriented. But another sentence that I had a hard time understanding, maybe you can comment on that. It's on page, let me see, 6. Actually, in the end of page 5, the last paragraph. And then it goes on to the beginning of page 6.
Okay, so the thermodynamic origin. Okay.
Exactly, the thermodynamic, yeah. Well, it's the last sentence when you're saying it. Some studies also have suggested that the conformation change, you're talking about the LNA, right? The introduction of the LNA, yeah. So the conformation chain induces a negative change in the entropy variation. And then I had to pause and think a little bit. Well, first thing, conformational change, you mean by just changing the tautomer because of the introduction of the LNA, if that's correct, what I'm thinking. And then maybe you can state in a more direct way. So what is, because it becomes a change of a change. It's a negative change in an entropy variation. So what is exactly the final change? You know what I mean? So
yes,
I was just thinking a little bit, it was like, what is she trying to say exactly? So
no, the thing I'm trying to say is LNA is going to give a change in the conformation of the helix. So the neighborhood is going to change conformation.
Okay. So it's in the conformation of the, it's the actual conformation of the double helix. It's something that's more localized to where the LNA is inserted.
Yes, because it is suggested that due to LNA insertion, you have a change from B DNA, from completely B DNA, for a more A DNA conformation at the neighborhood. So and some studies suggest that this change in conformation have a change in the entropy variation.
Mm-hmm.
Okay, I will correct the sequence. You're right.
Yeah, I was, because even this conformational change was not really clear to me. I was like, is she talking about? I actually thought you were talking about, because you're introducing something that's in only one tautomer. You're actually talking about the tautomerization that happens with the bases.
Yes.
And I was just starting to thinking, what's the time scale of the tautomerization? Can you actually prove this? And maybe it's something else. It's just the way it's stated.
Yes. Yeah, I will correct the sentence to be more clear.
Okay. Okay. No, that's fine. Okay. In the methodology section, it's just also something related to the writing. But you can correct this very easily. I would, instead of saying thermal unbinding of nucleic acids, I would just say unfolding or maybe denaturation, right? Because
yes.
Yes.
I have to make this here.
Okay, then, I mean, it's section 1.4.2, where you actually explain the PB model. I found it very well written. Yeah. I mean, I have to be honest, I don't understand it completely, but I think you did a wonderful job in trying to explain it to me. So I found it extremely well written, and I really commend you on that.
Yeah, I tried to, I opened all the equations just to be clear that I was not missing out anything. So that's when I actually wrote this.
Yeah, and I mean, it probably was a great exercise for you too, right? To explain these things when we really understand them. Yeah. Well, my advisor once told me that if you really want to know something, you need to teach this, right? So it's kind of like this. Yeah.
We have a teacher that he says, if you want to explain something, try to explain to your grandma. And if you can explain to your grandma, you understood.
Exactly. That's the idea. Okay. So one thing, at some point on page 9, at the end of page 9, you talk about cooperativity, right? And in thermal unfolding. And if I understood correctly, and I just want to comment on this, if I understood correctly, your model doesn't seem to take into account cooperativity. Is that right?
Actually, a little bit. Some of the models, I think that discussion is not well written, to be honest. But some models, they tried to do this thermal denaturation of DNA without including this cooperativity. What I mean with cooperativity is, if I break this base pair here, it's more probable that this next base pair is also going to break. So I think this is actually not my thing. This is inserted in the model through the stacking potential. So I have a potential that binds every single base pair in the stacking. So That's the cooperativity term.
I see. Cool. That's cool. Okay. Okay. So when you get your first paper, I mean, which I think is very well done. One thing that I actually found missing, I would say missing, but actually while I was reading, I wanted a little bit more information. And I know that when we do these things, we reference the information, and I mean, that's it, right? But I actually found myself thinking about what exactly are the sequences that you have used. And I think Luciana touched a little bit on that, right? So a little bit more information in terms of the sequences that you use to parameterize
You mean models. Which kind of sequence that I need?
Exactly. So how many of what length are they somehow biased to a certain sequence motif? Or, I mean, if you had more, is it better? You know, things like that. A little bit more information.
This is better discussed in the first paper that Gerald drafts about TEF, right?
Oh, okay. Okay.
The program.
The prior.
I can't clarify this, yeah, but we basically try to find it. Yes, sorry, just
okay.
The cable, but we are trying to clarify this. We need not so long, so not so extensive sequences, but also not so short. So the model needs at least 6, 8 base pairs to start, and then goes until 100. But I never tried anything with 100 base pairs, so just can be more clear about it. And it also cannot have any motif different than the duplex. So, for example, roads or anything, because otherwise we need to model this as well.
Oh, okay. Well, that's interesting. Yeah, that. So in terms of the in page, let me see. Well, in the methodology section of the first paper. So when you actually talk about the sequences used, so say that use 18 RNA duplexes at 4 different sodium concentrations, right? So, and also this is for the low salt, right? Sequence data set. So for the high salt, you also comment on the fact that you have some overhangs, right? Non-self complementary sequences. And it's just out of curiosity. Does it make any difference having non-complementary sequences? Okay.
Yes. Non-complementary sequences, they are going to have a different F in terms of the calculation for the nearest neighbor model. And for the Parabishop model, we need to restate which is the different complementarity and self-complementary when it goes this way. So they have this a little bit differences. So we try not to compare.
Yeah, I think one of the difficulties is actually the quality of the data set, the experimental data set, right? And I think Luciana touched on that already.
Yes, but we still treat them, and we still have acceptable results with this.
Right. Yeah. Well, you can actually predict the parameters that you want to predict in them very well. So I mean, even though the data set is not the perfect data set, the experimental data set is not the perfect data set that we like to have, it's good enough for the predictions that you want to achieve. So
yes, I was we were really impressed when we got this correlation with strand concentration, because we thought this was going to be completely different for every single F. And then when we got the results, they were really similar. So we were just like, okay, we wouldn't expect so much.
Yeah. So in your figure 6 of the first paper, I think, I mean, one of the biggest discoveries of this first paper is the fact that the terminal AU pairs in DNA are more susceptible to base fraying than terminal AT pairs,
AU.
right? In DNA, yeah, are more susceptible to base fraying than AUs, right? I think you can say that, right?
Yes, a little bit. We try
Okay.
not to write too much like,
Right.
I don't know, difficult sentences
Right.
to
Well, I
avoid so
mean,
many revisions.
if you, I mean, what do you think is the take-home message of your first paper?
What do you mean, the main message?
Yes, the main message.
So we can actually treat different data sets containing multiple strand concentrations. So I think
that's a big step, right?
Yes, the best solution, the best conclusion we could ever have from this paper is the Bishop model does not depend on the strand concentration.
On strand concentration, yes.
We assume it does that at first, but now we can actually say it, okay, it does not depend. And we can actually compare this different with DNA. So it's more an extension of the work that I did in my bachelor's.
Okay, so let's come back to the difference between this DAT and RAUs at the terminals, right? That it seems that at least it seems that DNA is a little bit more prone to base fraying than RNA, right? For at least for these pairs, these AU and AT pairs. Well, I have two questions regarding that. I mean, is there any experimental evidence for this in the literature? I mean, is this really backed by any sort of experimental evidence, or maybe another evidence that's not experimental, but has it been seen in atomistic simulations or things like that? And
yes,
and if you ever thought that, why is that? Why replacing the timing by a uracil, you would somehow weaken the hydrogen bond, if you can say
that. I'm just not replacing a timing by an uracil. I'm changing the sugar.
Oh, right. Okay.
So if I change the sugar, I also change the stabilization of the helix and the conformation. So I also change the way that the ions interact with the helix. So that's the main reason why AU is probably more stable than DNA.
So you think the AU is probably more stable than DNA because
low salt concentration.
It's a conformation for low salt concentration, exactly. It's a conformational thing related to this.
Yes, but I think it will depend on also in the neighbor. So you also need the neighbor to affirm that this is going to be more stable.
Right. Yeah, that was something else I was going to ask you. I mean, what about the neighboring effect, right? Because here we're looking at one base pair, but I mean,
so that's the reason I cannot say.
I see.
That's the reason I cannot say that AU is more stable than an AT, because it depends on the stacking.
Okay, it depends on the sequence, right? It's not just the AU, but you have the sequence motif. Okay.
Base pair is staying up there.
Right.
So perhaps if I have a CG, this is going to be more stable. If I have an AU, another AU.
Okay.
So it's just a lower Morse potential.
And I don't remember correctly, but I think that the papers that you're actually writing and you're working on has something to do with the neighboring effect. Or no? Or am I mistaken?
What you mean the stabilization?
Well, I thought it was. My question is, can you actually study this effect? So using your
of the neighbor.
Yes, of the neighbor.
Well, as far as I think, yes, because you have this correlation with the neighbor. So you have this stacking interaction. So you can say, okay, which base pair it's going to be it's going to do this. It's going to make this more stable. So that's I put on the figure that I wanted to show. So for example, this was really conflicts with the thing. So for DNA here, we have a bunch of CG at the ends. Can you see it?
Mm-hmm.
Although they have a similar opening, RNA is still opening more than a DNA because you have a lot of CG. So DNA in this case, if you want to say, it's more stable at the end. So it's also depends who is flanking. And I think also there is another figure of this opening. No, there isn't. Yeah, it's just for polyatodes. No, but is that this? I thought we had a different figure just stating different openings, but we did not have in this paper. But I think you can study cooperativity on this one. We said about flanking neighbors.
No, that's fine. So something else that I noticed in this figure, Izabela, and I mean,
Yes.
you can correct me if I'm wrong. I'm not sure if you can actually state that, or if it's somehow statistically relevant. And also, when you showed the movie with the different Fs, right? So this graph changes a little bit, but I was under the impression that there's a slight increase of the Morse potentials for both RNA and DNA at high salt concentrations.
Yes.
Yes. Can we actually interpret this like that? And what do you think it might be happening?
Actually, here we tried really not to discuss this comparatively because they come from different data sets.
Oh, okay, yeah.
So this difference here, so this abrupt difference that I see here from 621 for 1021 could be just an artifact of the data. So that's the reason we do not conclude like, okay.
Yeah, so the 1 molar salt is a different data set, right? And,
yes.
Okay.
And the different data set has this self-complementary sequences and everything.
Oh, okay. I see.
So this could be, I don't know, somehow impacting the final results. But we needed to put on this 1021 here. Just to say, okay, we did, and we have it. Does it make sense?
No,
but we cannot
it
compare.
makes sense. Yeah, it makes sense. It makes sense. Yeah. Okay, so when I finished reading your paper, two things came into my mind. The first one is we actually studied hairpin once. It was the DNA aptamer. Gerald collaborated with us in this work. And I was wondering if you could do the same thing for hairpins. I mean, can you actually get the TMs, if you can, the TMs measured for the hairpins, and you can parametrize. I mean, it would be interesting to see if there's differences in the internal loop and how differences of the internal loop maybe actually change the stability of the double helix, right? Or something like that.
Actually,
is that possible?
When I got into the PhD, my first thought was work with hairpins. I wanted to model them. I wanted to adapt the model for hairpins. But Gerald didn't. Actually, he said like, okay, but it's maybe, I don't know, because it's a different problem. There is a paper that actually supposes a potential for this hairpin. So it considers everything as a double helix. And then at the proximity of the loop, you have somehow a collision potential. So you have a different potential, because this is like, it's binding, right? So I cannot assume this is
Eu acho que ela caiu.
Alô, Izabela? Oi, Isabela. Just a minute. Must be a problem with her phone.
Yeah.
Okay, she lost power with the phone. and the microphone on her computer is not working for some reason. So she needs the phone. Just let me talk to her briefly. I would suggest you make a brief break, about 5 minutes. What do you think?
Okay.
Right. So we are back in 5 minutes.
Hey.
Hi,
Hey.
Izabela. Hi.
I'm sorry, Anderson.
No problem.
My phone turned off, but I can continue from my spot. Yes,
Okay,
just let me
so
share my screen again.
I think we were talking about the
Yes.
Capability of using the model to describe the stability of hairpins.
Yes. Yes. I already started this project, I think, in 2014, 15, with Eric, another student. He also wanted to model hairpins, but then they started with bulges and they could not finish the hairpins. So that was also a problem for me to take the problem for hairpins, because Eric wanted to finish. But I think from this paper, I can also send to you if you want to. But it uses a different potential at the proximity of the loop.
Mm-hmm.
So from this, you can maybe, it's like, I do not remember the word in English, is cisalhamento.
I see. Yeah, I don't remember either.
Sharing.
I think Jared knows.
Oh, okay.
It's sharing.
Sharing, yes. Yes, this scissor movement close to the loop. So
Okay.
it put on the different potential heat there and is assume a potential for the whole loop. But from this model, it's also using the Parabishop model, if I remember correctly. So we had to implement another potential. But this is I don't know, it's the future work I wanted to, but this I didn't have time, or just like I started to work with LNA because that was the thing we needed. But I think it's possible to model the hairpin, but it's a more complicated problem.
Right. Well, yeah, I think it's very interesting, and because this is the first thing that came into my mind when I read your first paper. I mean, can we apply that to hairpins? And then when I read the second paper, and why we were interested in hairpins, because we actually found a DNA hairpin that it binds at nanomolar affinity to one of the proteins that we studied. The protein is evoked in a disease, so maybe this could be used as a treatment or as a tool somehow of intervention, or even for research, right? As a tool for research.
Yes,
and,
I actually, for this paper, using this data of RNA, I did an opening for just the duplex for EGO 2, I think it's EGO 2, the hairpins, that interacts with EGO 2. But I cannot do the full hairpin, unfortunately.
Yeah, but one thing that I was going to say that by the end, but one thing that so the first thing I thought is, can we do that for hairpins? The second thing I thought is, I mean, it was very interesting the way you actually predicted the insertions of the LNA. I think that was fantastic. And if there is a way of actually stabilizing the hairpin, because one of the problems when you work with hairpins, especially with so short sequences, is that they're actually heterogeneous in terms of conformation, right? They can be open, they could be closed. Some of them are thermodynamically more favored, but you do have an equilibrium, a conformational equilibrium. And so I was wondering if we could actually stabilize the closed conformation of the hairpin, and maybe this would make it bind even tighter to the protein, or this can change the activity it has in vitro in this biological context, right? And I think that's another application of your work.
Yes.
I mean,
I think we could do that using the Parabishop model. So we could use this average openings and thermal stability, but we could also use the nearest neighbor model using I don't know if you're familiar with Vienna, the secondary structure predictor. Also using the secondary structure, so the Gibbs energy of the secondary structure, but it's like I cannot just discuss more than this. Very deep.
Okay. No, I see. well, moving on. Okay, so then we get to the second paper. That's just out of curiosity, Izabela. I don't know if, I mean, if you actually did this, but I mean, when I was reading the paper, I was, getting to a climax, and I was like, okay, maybe she tested the design probes, and that they were actually better probes, or the detection limit was lower, or something like that. Or maybe this is for the next paper. So it's just out of curiosity. I mean, it would be interesting to see if the newly designed probes were able to detect the predicted oncogene. Oh, okay. Did you guys test it? Oh, they didn't.
No, we didn't. Also, Luciana asked it, but we didn't. I don't know if Kira still already did, but as far as I know, she still didn't do it, the binding with the targets. So I don't know if she can comment on this, but then you could actually measure the probe efficiency and everything.
Yeah, right. And see if the actual changes you.
Yes, if the probes were actually efficient. Yes,
exactly.
We didn't.
Yeah.
We only had like the temperature prediction.
Okay.
But this is interesting for future works.
Well, this is very interesting. Yeah, this is very interesting. And one thing that actually Jared said, I mean, the how the model is the simplicity of the model is also the beauty of it, right? That actually allows you to test so many different insertions and modifications and things like that. So this is this is very powerful.
Yes. But the limitation we have is exactly the parameterization. So that's the reason we are still doing the whole parameterizing step.
So another thing that was very interesting in your second paper is the whole water thing, right? If you actually have water mediating hydrogen bonds between the bases, and so if I understood correctly, there's an NMR paper that actually suggests that you actually have a tightly bound water to the nucleic acid. And this is just suggestion, and just because when I read that, this is the first thing that came into my mind. And as an experimentalist, we're always trying to find an experiment. Can you actually do an experiment and test this hypothesis, right?
Yeah.
So in terms of bound water, I mean, if there's water mediating the hydrogen bond, you may see some difference in terms of how the TA values for the canonical sequence and for the sequence containing the LNA modification vary with increasing concentration of osmolytes, because at some point the osmolyte will displace the tightly bound water. So every time there's water involved in some sort of interaction, and you probably see a difference dependency on osmolyte concentration, because the osmolyte will shift the water. So it's just a way of maybe trying to look if this difference you see is actually related to the fact that it would have a tightly bound water or something like that. It's just
yes, I think that we could perhaps study, actually, I don't think we have experimental data for this, but if there is experimental data on different salt concentrations, we could actually do this, because at some point you're going to have competition between the ilex and the water to get the ions. So you could actually state, okay, there is water bridged. Indirectly say, okay, there is water bridged hydrogen bonds or not. But there's an indirect aspect. I don't know if we can actually do this with NMR.
I mean, you can try to do some measurements. I mean, it's not really easy, because every time because you have tightly bound water, you have water around, and you would have to actually measure things indirectly. But there are ways of actually. I've never done any measurement regarding water, but I know that there are some. And
okay, so it's
one we have to look into. It's possible. Yeah, it's possible.
Yes, because one on NMR studies is suggests exactly that, just to include water in the experiment. but then you need to consider every single molecule of water in the whole animal. So it's like costly, I think.
Yeah, I mean, it's probably what he did is he saturated the water. I mean, didn't see the paper, but for you to say that there's water bound to a site on a macromolecule, you probably saturated the water, and then you see saturation transferred to a specific site, one of the hydrogens. And then you say, okay, because I'm saturating the water and the saturation is being transferred to this hydrogen on the DNA or whatever, that's because water is really close to this hydrogen, so it's probably tightly bound and
so it's probably binding.
Something around that, yeah.
Okay, so is it still indirect?
It's possible. It's still indirect, but it's possible, yeah.
Okay, yeah, it's a good study also. Thank you for the suggestion.
No, I mean, it's just, and the last thing, I mean, since I already said, one of the whole LNA thing is, it's very beautiful. I think it's very interesting, and the application is very interesting. And I was curious about this bound water, and also, I mean, to the fact that so can you actually say that the hydrogen bond, it's getting stronger somehow due to the insertion of the LNA, or is it because you have a third hydrogen bond?
Yes, I get the question. I think it's a pretty tricky question, because this Morse potential is going to measure the efficient potential, so the effective potential. So I cannot exactly say, okay, there is a third hydrogen bond, without actually seeing structurally if it is possible to form one. Because perhaps this comes from, I don't know, there is a water stabilizing this site, or there is anything else. But the Morse potential is measuring the effective potential. Does it make sense?
Oh, yeah, totally. Well, I mean, Izabela, I think you did a wonderful job.
Thank you.
And I think you defended yourself very well. And with that, I finish my argument here. And I can only congratulate you.
Thank you very much for the suggestions and for the compliments. And thank you for coming to the defense.
Okay, thank you, Anderson, very much for your comments. And regarding the hairpins, as Izabela already told, it's an old project in the group. It's also a little bit like a hot potato. It just jumps from one hand to the next because it's a difficult project.
Yeah.
And I think what lacks perhaps at the moment is a good motivation. And if you have a particular interest, and I mean, we have collaborated in this before, but not in regard to this type of project, then that could be the motivation that is missing, that will make us move forward into finding a way to model this with the Ferreira Bishop model. So we definitely need to keep talking.
Yeah, sure. For sure, yeah.
Okay.
It would be a pleasure.
Right, so let's move on now to the members from our university. So we have, I don't know exactly who would like to start. Could be either Simone or Mariana, depending on who is fastest to say hello. Simone,
the first to speak. The first to speak.
Pardon me?
Hi, Gerald.
Hi.
Wait, sorry.
You have an echo.
Mariana.
Hi. I think Simone can start, right? I can go to the end. I'm from the program, so.
So technically, you are closer to the.
Yeah, I'm the closest one.
Right. Good. I think so. We could say about Simone, but I don't hear Simone. Maybe she's not in front of the computer right now.
I'm here.
There you are. Good. So let's let's move on with Simone.
Yeah.
Simone, thank you very much.
Okay. No, I would like to thank you, Gerald, and you, Isabella, for inviting me. And it was a pleasure to read and participate in some way of your job. And I have argued with Isabella quite recently in her defense of the qualifying exam. So I don't have many doubts. And some of my questions have already been asked by other colleagues. So I guess that will be easy, my part. Okay, one curiosity. Is this thesis format standard in bioinformatics program?
Is an alternative to do, because they say if you have.
I mean to put, for example, to put the results as papers attachment.
Yes, it's an alternative format. So if you publish 2 papers as the first author, you can just put on the papers and do an introduction. Because I needed to defend fast, so that's the reason I did them this way. Otherwise, I would write everything.
Okay, no, it's just a curiosity. Because in our department, we always discuss about this. And,
it's not allowed, I think.
Yeah. I'm still in doubt about what you mean about strength concentration. It was confused for me, but my colleagues asked you quite almost the same question. But what do you mean? It's I don't know, the numbers of strengths of DNA, for example, in the solution. What does it mean?
Yes, it's the number of strands that you have in solution. So if you have 2 duplexes in solution, so you have 4 strands. So that's the strand concentration. So if you do this in terms of mole, you have the micromole notation measurement. I think they measured this in the absorbency, but I don't remember correctly. Jared can correct me. But that's what I mean by strand concentration. Each paper defines as this.
Okay, thank you.
Makes sense?
Well, yeah, thank you. One other question is, you told me sometime, maybe you or Gerald, I don't know, that you are working in the 3D model, PB model for DNA. How is it?
Yes. What do you mean, the PB model? I don't know if I got the
which
question.
freedom of degree?
The 3-dimensional model.
Okay, the 3-dimensional model. No, I'm not working with the 3-dimensional. I'm just working with 1D.
Sorry, the 3-dimensional model was someone else's project.
Yes. Cheryl told me that we actually can treat everything else that we model until now using the 3-dimensional, but then we needed to model everything again. So this is not in the 3D model. I think would be like a good project to get a student on.
Okay. About the LNA section, we found the H bonds in LNA is it's stronger when compared with DNA. Did you make some calculations for RNA generated from RNA?
If I found any data from RNA format from RNA.
Okay.
Actually, no, I get the question. You mean adding RNA modifications in a RNA sequence?
Yes.
I found some data doing this, but it's very, very few sequences. So we cannot model is usually done in DNA, but I didn't see any melting temperatures using RNA. But if you have, I think it's doable.
Mm-hmm. But could you use some results that you find for the salt concentration problem to predict some, I don't know, to put some term, for example, in your model, to modulate the LNA from the RNA?
At different salt concentrations?
Yes, because in the salt concentrations problems, you calculate DNA and RNA, right?
Yes.
In different concentrations of salt. And you found some different results, some differences of DNA and RNA in this problem. And I'm asking if it's possible to get these informations and to do some kind of reverse engineering, like Weber like to say, to modify your model to calculate the LNA, sorry, generated from RNA. It also depends of the melting temperature experiments.
Well, if I got the question is like if from our data from RNA, the RNA project, we could actually apply this on LNA.
No, apply the results to get some information from your results from the difference between DNA and RNA, and to improve your model to do prediction
yes,
in the
I think,
LNA case.
Yes, I think the best result that we take from RNA, the RNA project, that is transferable for LNA is that the melting temperature will not depend so badly on the strand concentration. So that would be the result. But I cannot say, okay, I have LNA DNA sequence and I want to predict some LNA and RNA. Otherwise, as far as I know, we would need a completely different parameterization. They could be relatable, but I cannot affirm they're going to be the same. Does make sense, I think.
Yeah, can be. Comparing your rate epsilon graphs for the salt concentrations problem, the LNA one.
Yes, the first LNA.
Okay.
I think it's this one. This first one here, or the ones that I got?
No, the other one that there is a epsilon rate.
This one.
Yeah. If you compare this one with the LNA case, we can see that there is one order of magnitude of difference of the average. Here you have average opening in order of 10 to minus 2 nanometers. And in the other cases, for the LNA, this is the LNA or RNA?
Here I have a full canonical sequence in blue, and in red I have the modification.
But this is the salt concentrations problem or LNA problem?
No, this is LNA. You meant the salt concentration, but the RNA.
Yeah, I want to compare the both.
Do you mean? Wait, I'm sorry, I'm confused. You meant this one?
Yeah. If you look to the epsilon x, we see a difference of one order of magnitude of opening between this problem and the other one.
Yes.
Can you explain this difference? That this have some meaning, some physical meaning?
Yes. I'm still, actually, I'm a little bit troubled. What this result is you got from molecular dynamics. So they do the distance between each base pair for each base pair. If I got this here, and here we have like it's comparative in a qualitative way, but it's not really strictly compared. Yeah, here we have like 10 and 10 is here, but it's not too different still. We have a more wider opening.
Okay.
So it's the opposite. If I have this to open this side, here on our results, they have to be like less open. Does that make sense?
Yeah, but I mean about the numbers,
Yes. Here is like angstrom, and here is not in angstrom. Okay, that's the thing.
Yeah.
But this is angstrom.
Yeah, this
It's
is
just nanometers. Is that?
But my question,
elevated.
when you compare this opening from in this problem with the LNA problem, we have a difference of 1 order of magnitude in these measurements.
An LNA problem.
Yeah, when you compare the both, the salt concentrations problem with LNA problem.
Okay, so here I'm at 10 nanometers, and then for LNA, I am at I am also at okay here.
Then yeah.
I got. Yes. I think this is going to open last, but for that figure on the RNA, this was done in 300 kelvins, and this was done in 150. So this is going to impact the opening. So usually when we set the temperature, the reference temperature to calculate the average opening, we try to find the ones that you can actually see something. So if I put on like 300 degrees here, it's also I don't know, it's more a short sequence, it's a shorter sequence, and it also is going to melt. So if I put on 300 here, they're going to be separated already, so I cannot see any binding. So that's the reason of this difference here.
Okay.
So it's very much the calculate temperature that we use it.
I got it. And one last question. If you have some, I don't know, some bad experimental results of melting temperature, for example, are you able to identify that these results are bad by, identifying in your parameters, for example?
I had this problem with this data for RNA, because when I did this for the nearest neighbor model, when I got the parameters, I got completely different parameters. So I could see like, let me show an example here. For example, I could see this error bars going like way too big. So the minimization is kind of trying to find a minimum that does not exist. So it's assuming anything. So I have this very, very different deviations from the other results. So then when I go back to the data, to the raw data, I see there is some mistake. So for RNA, I had to do this over and over and try to find the errors in the measurements or the errors in typing and things like that.
Okay, nice. Okay, Izabela. So congratulations, you did a lot of work here, and very, very nice work. And so
thank you very much.
Congratulations for you and for Gerald as well.
Thank you very much, Simone, and thank you about the comments, and thank you for coming.
Thank you, Simone. Simone is one of the persons you can always ask to come, and she's super patient with our work, always trying to get into the details. And the question you ask about the difference, I mean, it's an order of magnitude difference. It's an important one, because what we do basically, we just want to have an idea, a qualitative idea. So because we know that we cannot take this Y number very seriously, because it's a two-dimensional model, not a three-dimensional model. But you're absolutely right. I think we need to take a lot more care with these numbers and present better explanations for them.
Yes, I haven't seen this difference between the measurements, because there were two different words, and then when you show, just like, okay, it's the temperature. If I did this in 300.
Yeah,
I
accepted this that Jared told. It's something that it's only a qualitative information, and sometimes it's I try to find some meaning.
We
you can just see regions prone to something. Now we just need Mariana, I think.
Okay, thank you, Simone, again. And now let's move on to Mariana. Mari, are you there?
I'm here.
Hi, good.
Hi, Gerald. Hi, Izabela. Good to see you again.
Good to see you.
Hi, Kira. Nice to see you too. Well, I think Simone and Luciana, right, and Anderson, they got into the point with Izabela. I was in all steps from Izabela PhD, right, Izabela? And the first thing I want to say, it's very important, because I don't know if everyone is aware here, but when Izabela applied for our program, she applied for a master's degree. And because of her argution and the way she discussed her results, and I think everything we saw in her, and she already had papers published, right, Izabela? And she was offered the opportunity to do her PhD, right? And it's very beautiful to see that you did this work, like it's amazing. I think it's really nice. You have new work. You didn't do only the things you were doing before. You have these papers that you recently published. You had the opportunity to go to Kira's lab, and, you know, as Anderson mentioned, you have this international experience which is very, very important for us, right? It turns you into a different person in some ways. And you wrote your thesis, and also you qualify in English. So, I mean, this is really good, and you having your PhD defense in English. So this is great, I think. So it's good to see that you are like this. You are a great student, and you were my student in many classes. So I have nothing to say about that, but only that you accomplished, in my point of view, all the things that we expect from a PhD student, you know. And as I was in your qualification before, we talked about a lot of stuff, me and Simone, and I could see the differences and the things that you change, and the way you try to bring other stuff together and make your thesis more clear, because it's very hard for us that work in a different area to make it clear for biologists and the person in bioinformatics, for the physicists. So everything is very complicated. And I think you go in there, you know, and to help you a little bit with that, I would ask you, like, can you describe in a few sentences what is the idea that binds your thesis together?
Well, I think thank you about the comments and the compliments. Thank you very much. Well, I think the whole thing, the whole glue, putting everything together, is the Paradigm model, of course. But LNA was a completely different project, not completely, but different from what I was doing before. Because RNA is a little bit of, I don't know, left over from my bachelor's. So LNA, it came with together with Kira. So Kira needed the results. So perhaps Kira is doing the LNA part and the bachelor's is doing the other part, but everything comes from the Ferreira model. Although I have different results using the nearest neighbor model, but the thesis is pretty much the Ferreira model.
Great. You know, like I missed something in your thesis when you're writing. Maybe you know that LNA now, because of everything that we are living, it's very popular these days, right?
Yes.
Because people use LNA in these probes to reduce background when you're doing qPCR, right? So it's very common, and I think many companies, they sell these modified like sequences and with LNA, right? And when you buy this, they give it to you, the melting temperature. They give you like melting temperature based on the locked base and everything. Did you try any of these commercial primers that they sell, these probes that sell? Try to compare your results to the ones that they give, like they say, are the results that we have, and you compare with the way you are doing your analysis, and you know where is the difference and how this affects. So mainly I'm asking you if you're looking into what people are doing and how your work affects your field, the field that we're working on.
Yes, I got your question. No, I didn't. Unfortunately, I could be more cited by now if I did, if I compare it, the sequence they have. But I don't know if they disclose the sequences. To be honest, I didn't look at, but I can give a look and try to do this. This would be a really interesting thing, just to say, okay, we are better than them. I don't know, 20% better, 15. Is this still a better result? Yeah, but I didn't. I think Pamela is working with SARS-CoV probes, and I don't know if there is anything that is modified with LNA. But if it is, it's interesting to look. Yes.
Great. Izabela, just a question, maybe because I didn't understand completely, but in your first paper, you tried to solve the problem that you had with RNA, and like your experimental data, it's very confusing. I saw your first discussion, and it's very interesting. Actually, that's how it goes when you work with biology. You know, everyone has one different parameter, and the measurements, it's very hard. So when you figure out, when you put in logarithmic groups, right? And you saw, like, and you wrote that on the thesis and on the paper, that surprisingly, there was no difference between these groups. When you apply the same thing to DNA or other probes, it works in the same way. So how does it work when you apply in different models?
Actually, we didn't apply it at this grouping because we wanted this to treat exactly this problem. So we didn't apply. But if we have something like DNA scattered over a range of strand concentrations, we could do the same. And as far as I know the method and the model, I think the result will be the same. We would still see a little dependence on this grouping, because the model shouldn't depend on the strand concentration.
Yeah, that's what I thought, that it shouldn't depend on that. Great.
Yes, we are just assuring that it does not depend. So that's the reason we could do that, and we got this, I don't know, good results or tricky, because we were expecting something completely different.
What you were expecting? Just
a completely mess.
Really?
For every F, yes, for every F, we're expecting like, okay, this is completely different. This does not work. So Gerald said like, okay, try every single F and see what is the result. And then when he saw like everything was almost the same, he was just like, no, it's not possible. I have to see. And then he checked everything and it was the same. So that's the reason we did all this, I don't know, this tricking with the regressions and stuff.
This is very interesting. You know, I don't record reading this in your thesis, but I think it's very interesting for you to describe this, like what you were expecting and maybe why you were expecting it to be a complete mess. And I mean, not just surprisingly, but also to, I think science had this way. Sometimes we don't try different things because we think, oh, this is not going to work, or maybe it's not, we're not going the right way. And maybe we need this motivation when we are reading, like someone teases and we say, you see, sometimes you need to try this. And maybe when you try, it gets you in a good place, right? And you get,
yes,
like you now are smiling very like happy because you are not expecting this kind of result.
Yes, I think in the qualification exam, I didn't put this because the same way as we did in the paper, we tried to sell the best dish possible. So we you don't put like, okay, I wasn't expecting this at all. You just flourish things and say, okay.
Yeah, Professor Simone, she raised a question and she cited this paper on Nature that they said importance about you sharing negative results, you know, and I think it's really important actually. I'm saying that because the paper is different from the thesis, you know, like I think the thesis is something that comes from you, it's your work, it's your book, you know, the name
yes.
of the page is yours. The paper is different, but I think it's very interesting when we read the thesis and we have these insights from the thesis, you know, because
you have the problems that we have.
Yeah,
we don't explain this on the paper.
Yeah, but not the problems at all. But, you know, like when you're testing hypotheses, you say, I was expecting something like that because of this. And it's very interesting to see that we actually, as you say, Gerald would not believe in you. So he went there and checked all the calculation you did probably. But this is really nice, you know. I was actually very surprised when I was reading the paper and this is really cool, actually. So you need to go into a problem with RNA that you have all these messy literature results to see that actually you can solve this problem in a very easy way, right? Not easy, but you know, you have a way.
Actually, at first I wanted to really discard the whole data and say like, okay, we are not doing anything else. I was fighting with this data for so long, and everything was like really, I don't know, disappointing. And I say, okay, I'm not doing. And Gerald said, like, we are going to find a way to treat this. And we did. It's a little bit of MacGyvering, but it works. It works. I don't have a word for gambiarra in English.
Izabela, I think I know the answer, but what would you think that could improve your work?
In the way of what? Which part?
I mean, you got at some point, right? And probably you have some thoughts about what is missing for you to do with your results or your work, what you would like to do and you were not able to do it time, or maybe availability, or I mean.
Actually, I wanted to do many things, but I didn't have time. But there is a lot of LNA data for different salt concentrations and also mismatches that I wanted to treat. But I was doing this a few months ago, and then I got stuck with different things. But I wanted to finish this if I had more time. And also for RNA, I think it's a closer, but we also could try this for different data that we could not deal before. So I think there are many options. I don't know. There are more things to do. The hairpin stuff, I wanted really badly to do the hairpin stuff. But
I think it's very, I had this discussion once with a colleague. sometimes people at their written like PhD document, they write like perspectives, and I always find this funny because you finishing your PhD, there is no perspective for you like in your thesis, right? But I think it's very interesting for you to write. I miss that in the writing.
Yes.
Where you are going with your work, you know, like what would have improved this work, and what is the impact that you are going to have in your field, you know. Maybe
Yes.
it's in together with what Simone was saying, but having together the limitations of your work, where you can go with this, and where you would like to go, you know. And I think we have this, especially with LNA, I see with this biotechnological world growing, and all this popularization of what is a probe and what is a qPCR, you can sell it in a very interesting way, you know.
Yes, sure. Yes, I should have written a perspective. Yes, you're completely right. I was just like so rushing so badly with everything that I completely forgot the perspective. But I will write one, I think, just to, because I'm going to work with something I think completely different in the postdoc. So that's also a point.
What are you going to work with?
They work with single cell RNA data, and they model interactions between transcription factors and different diseases, and also cell-cell interactions and stuff like that. So I think it's different than I put on here.
I think you're going to do well with that.
Thank you.
Okay.
I hope so.
So together with what I was saying before, I think it's interesting, and I want to hear from you, and maybe you can add that one line or two at the end of your thesis. What do you think are the long-term contributions of this work that you did during your PhD?
What do you mean the long terms?
I mean, what do you think this is going to impact, like the field you're working in?
Yes, I think we broke the model, so we can trade more things, and especially with the probe that you commented, we can also treat more realistic problems. We could also, like, instead of searching someone to collaborate with, we could actually be asked to collaborate, the way that happened with Kira, for example. So I think there are a lot of better perspectives as we build all this Terabishop model parameterizations.
Okay, Izabela, I'm happy to hear from you, all your thoughts, and see the way you were discussing your work with me and your other colleagues. I wish you a lot of success in your future days and your career. I think you are on the right way. I'm sure Gerald is very happy with the work you did, and you know, it's really good I will say that again, because it's really good to see you from the beginning and where you are now, and it's really nice. Congratulations, Izabela.
Thank you very much, and thank you from the start. Thank you very much.
Thank you, Mariana, for the comments, and it's always goes down to the point. Sometimes we don't have just the data to do the things. We want all the data are not available, right? It's always comes down to that. So I will now, so this concludes the internal members. I will now open for Kira to make any comments or questions she wishes to do. Hi, Kira.
Yes, hello. Can you hear me?
Yes.
Just a moment. My Zoom is tricking me today. Oh gosh. I'm very sorry. I don't see anything, sorry. But maybe I shouldn't see anything. Anyway.
We see you.
Okay, that's good. I don't see any, it's just black screen, but I'll try. So Izabela,
I think you can, I don't know, roll left or right.
I see your thesis.
And or use Alt Tab.
Okay. But I mean,
I can also stop sharing, and then you can see the
thing. Thanks. So I also want to say congratulations on your work. I think you did really great, and I've seen it in progress, and I think it developed nicely. I want to say that I feel we learned a lot from working with you. You really contributed strongly to our research. Before I started collaborating with you and Gerald, we pretty much used kind of random approach to design of our probes, as everyone does in our field. We just put some modification somewhere and screen. So I think for everyone, it has been very great experience to get more in depth and see that the modification position really matters and how exactly it does. So I appreciate this a lot. And I think that's the direction that we will pursue, because it makes much more sense compared to the random screening that we have been doing, along with, as I said, everyone else so far. I will cite my old-time supervisor, collaborator, who said that problem of nucleic acid interaction is forever problem. It will always be there. It's a task that is so appealing, so important, and I mean, it can be always improved and get better and better. So I think, yeah, we as people working with nucleic acid detection and predicting the probe binding, all of that, we will always have some tasks to solve, because it is, genomics is a huge field, right? Yes. And there are so many.
Work. Yes.
There is the work for definitely our career left, right?
Yes.
I have a few questions that I wanted to ask you.
Yes, please.
And one of them is, if you could please tell me a little bit about the limitations of the model that you are using.
Yes.
So it has strengths, but what are the main limitations of this model?
Yes, as Gerald said before, we are considering a one-dimensional model. So we are not considering anything ahead from the sequence. So this could be a limitation. So if we have a different, I don't know, an important impact from the shear of the helix or from the spinning, this could be a problem. Also, we have this the whole parameterization limitation, that because we need to parameterize things, because especially with you, sometimes we have you wanted us to do some data, to run some data, but we do not have the parameterization, so we need to run this again. And if we do not have enough data to parameterize, we cannot predict anything with certainty. So I think this is the main limitations of everything. And it's also a model that uses two simple potentials. So even so, we have good results. Some things are still ahead from us. For example, secondary structure or anything else apart from the temperature.
Yeah, I see. I also actually want to, please disagree with me if you feel like that, but I think that linking to exclusively TM as an experimental validation is also limiting it, because this is not the only way to measure nucleic acid binding. Again, please disagree. And we always try to verify our data by a second method, but in this case, we solely rely on TM, which is also, I think, it's not sort of, we believe it's correct.
Yes, we try to have a parameter to rely on. And the temperature, I think, is the easiest way to do. Yeah. But I don't know.
That's the only one, right?
Measurements. Yes.
But that's the only one. And it could also introduce some
bias.
Some bias, yeah.
Yes, I agree with you, yes.
So there has been many comments. I don't want to take more of the time. I want to say again, but it's been really, it became a curriculum here. When I teach advanced biomolecular chemistry, I introduce your model actually, because I think it's so important.
Nice.
It became a part of it, yeah. So I think it's
nice.
important. And again, moving away from screening methods towards more rational approach, I think it's definitely the future of it, because it's so useful. When it works, it really makes a huge difference in our work. So I only can say thank you for this experience, and I wish you all the best in your future career.
Thank you
So
very much for
I don't
the
have
reception and the comments. No, you can say. I'm sorry. I'm just thanking.
I think I'm done.
Okay. But anyways, thank you very much. The work could not be complete without you. So I think most of our paper came from the exchange. So thank you very much for the whole contribution and for all the advice and everything. Thank you very much.
Okay, thank you, Kira, for your comments. I'm the supervisor, so I'm always the last to speak. First of all, thank all the board members for their comments. And I need now to thank a few people very specifically. I will start thanking Kira for making it possible for Izabela to spend 7 months in Denmark during the beginning of the pandemic, right? This was so, I mean, Izabela just arrived in Denmark and then they went into lockdown, which was at the time totally weird for all of us. Right?
Yes.
But eventually things worked out pretty well. So thank you very much for that. And I have to thank a lot to Mariana, because she was on the selection board that interviewed Izabela, and they saw her potential to move her on straight to the PhD program instead of doing a master's first. And I think this was transformative. I remember 1 or 2 months later in the corridor, I met with Mari and I told her, I have a new student. Izabela is totally different from Izabela from before. This was a phase transition. And the second phase transition was when Kira visited us and Izabela met Kira and something happened, which I don't quite, I'm unable to put my finger on, but something happened, and that happened a second phase transition in Izabela, and which is the result of everything that you are seeing, and then the conclusion, the hard work that she has put into the 2 projects. It's not fair to say that, Izabela, I will disagree on one point with you. It's not fair to say that your RNA work was a leftover from the previous period. It was a leftover problem.
Yes.
It's something that we wanted to do, and we always, we have to do the RNA stuff. We have to do the RNA stuff. And then eventually we said, no, we're going to finish this. And then is when we had to solve that problem with the concentration, right? I mean, you started this last year, basically. And that's where we had all the interesting surprises of the results. So these are the 2 main things I have to say. Everyone in the group who also interacted with Izabela, well, I mean, the group interacts a lot, sharing ideas and tricks and data and so on. So everyone in the group has also their hidden contributions to everything that just happened. So it was a pleasure. It was super nice. I mean, Izabela started, when was it? 2015, something like that. 14 in the group. She basically had just entered as an undergraduate student, and she basically went up to know
I'm the
everything.
oldest. Now I am the oldest.
Now you are. And it was super interesting to see how she progressed in her PhD, which she did in record time, and securing a job so early in the career. I mean, it's a postdoc, but it's a job, which is another fantastic thing. So I hope you're going to be very successful in your new job. It's a pity that you are leaving, and now that you're being so productive, but that's how it is, right? So, okay, now we will now, I will ask the board members, I will open a breakout room here. And,
Professor, já está aberta a sala simultânea, eu vou transferir a banca para lá, tá bom?
Ah, é? Tudo bem.
Ok. Isabela, enquanto isso, aguarda aqui na sala principal, tá ok?
We are being transferred to another room where we will deliberate about what we're going to Muito bem, voltamos. Izabela, cadê você? Muito bem. So it's my pleasure to congratulate the new PhD in the group. Izabela, congratulations. You have to open your phone, your microphone. It's not being easy today. Izabela's iPhone.
Hey, I'm sorry.
Okay, so again, congratulations. You were approved. You are now
Thank you.
a PhD.
Thank you very much. And I'm sorry about the whole technical. Thank you.
It's so weird doing this over Zoom, but anyway.
Yes, I was just like, okay, I don't want to do this by Zoom. So I still did the whole outfit and the whole thing.
Good, congratulations.
Thank you very much.
Thank you, everyone who stayed to the end and this long session.
Thank you very much, everyone, for the comments and for the compliments. Thank you very much.
Good, so I will now stop the recording. And I think we can also stop the YouTube. I think I don't have the power for that one. That needs higher powers than mine.
I think that needs Sheila.
And I have an important question.
Yes?
Sorry, what?
How about the celebration?
Yeah.
Yes, celebration.
It will be with a very good beer.
The beer is in the fridge.
Yes.
Waiting for Izabela to arrive.
Yes, I'm arriving tomorrow. Yes.
Nice. I will be waiting for the invitation, right?
Yes. Yeah, Anna.
Hello, Anna.
Hello. How are you?
Fine. And you?
Good. How you doing?
I think he's going to bring the beer to show to us.
Oh, wow.
Perfect. I printed the screen and then I will send you.
No, I don't have one of these beers. This is like the tradition of the group. I need one of these.
Thank you all. Good to see you. Izabela, parabéns.
Thank you. Thank you very much. It was really nice.
Amazing.
Thank you. Thank you everyone for the patience to stay here until 1 p.m.
And the external members, you have now to go to the say thing and digitally sign the form there, okay?
Okay. Can I leave the room or I still need to?
Parabéns, Izabela.
Obrigada, Anderson. Muito obrigada.
Parabéns ao Jared também, a Ana, né? E a Kira. For the...
Jared, você vai mandar champanhe pelo Uber? Pelo Uber Eats? Tô esperando aqui.
Meu Uber é mais caro.
Vamos ver se você vai vir mesmo, Izabela, amanhã ou depois? que você pensou aí?
Eu tenho, em tese, algo marcado amanhã, mas aí eu combinei com que ou a gente vai amanhã, mais à noite, ou na quinta. Vocês preferiram, eu não vou atrapalhar, mas eu vou para BH amanhã.
Para mim seria melhor.
Então tá, amanhã tem. Tá certinho, combinado.
Ok. Legal, eu vou ver alguém, pessoa.
Acho que deve ter uns 2 anos que o Gerald não me vê. Thank you, Eric. Thank you very much.
2 anos mesmo.
Tem uns 2 anos.
Dinamarca, te ver pessoalmente.
Oi?
Esse que foi para Dinamarca, não te vejo pessoalmente.
Exatamente. Não, desde quando o Kira foi embora. Eu não fui mais no Rio.
Muito bem. Então, vamos encerrar aí, gente. Muito obrigado, Kira.
Beijos, tchau, Ian.